basically the plant disease detection via image recognition that yesterday we saw in the practical class, right? So it's going to be similar content with some few more examples on importance of diseases and how like the science overcame the disease incidents and how we produced resistant varieties. We may not look at the irrigation or fertilizer planning. We will mostly focus on plant disease detection. Right. So I want to start with the father of botany. Right. So he is Theophrastus. He is the father of botany because 300 years before Christ was born, he wrote two books. One is The Nature of Plants, another one is The Reasons of Vegetable Growth. So the disease is not a new term, it's very old term. It has been mentioned from even before Christ in the books by Aristotle. So Theophrastus is nothing but a student of Aristotle. Yes, Aristotle. Yes. Yeah. So in The Nature of Plants, yeah. So he has mentioned about plant anatomy, morphology, and wild and cultivated woody plants and cereals like wheat, rye, and legume plants like our chickpea and soybean and medicago, other legume plants. And in the book Reasons of Vegetable Growth, he has mentioned how we can propagate plants using seeds, right? So we have seeds, you plant the seeds, you'll get what are the plants that seed can grow into. And also he has mentioned grafting as well. So the grafting is very interesting a technique. So recently you might have seen too many grafting techniques. So they graft mango trees, right? Different mango trees, different flowers, different vegetative parts. Can you tell me why? So there is a relevance to disease resistance. So have you heard this term, rootstock and scion? So the reason, relevance of grafting in case of disease resistance is that if you have a rootstock which is resistant to plant disease, right? So you need only the rootstock where you will have, you can get a susceptible, sensitive plant, scion. And then if you graft this scion, which is susceptible to a particular disease, this scion will become resistant because the factors, proteins will move from resistant rootstock to scion and make it resistant, right? Because it's unfortunate many times that more yield producing plants or producing variety tend to be susceptible to diseases, right? So the more better variety which produces more mango or more rice yield, they tend to be more susceptible to disease. So in that case, you take the scion from the more yielding variety or better variety, taste, whatever, and then you graft to a resistant rootstock, then you're going to get a resistant new variety, not the new variety, new plant with the desired traits like your sweet mango or whatever. So far nobody has done grafting in rice or in monocot. So doing a grafting in a monocot is not easy. So very recently, Professor Pallavi Singh, she was my senior in NIPGR where I did my PhD. So she did postdoc in UK, Cambridge University, where she performed grafting in monocot. And for that work, she has become very famous, and she got a permanent position in that university. Now she is analyzing how the resistant molecules move from rootstock to scion and how they provide resistance to the scion. Yeah. So still the grafting is not new. It was mentioned in a book written by Theophrastus 300 years before Christ born. So none of them are new. We know what is disease, we know what is propagation, what is grafting, and also he has mentioned how environmental changes affect a particular disease output. So yesterday we briefly discussed, right? So in order for any disease to occur, right, to attain its maximum potential, it needs favorable condition, right? In case of that fungus that I worked on, dry root rot, it needs dry weather, less soil moisture. Then only it can cause 100% disease severity and cause 100% yield loss, right? Yes. And also propagation of cereals and plant disease and plant death and the taste of plants. So several plants. So we might wonder, right, like how we cultivated, how we did breeding to get like wheat or rice with a better production, right? So all works were done even before the Christ was born. So that is the reason that we give him the title of the father of botany. And point to be noted, he's a student of Aristotle. So obviously he'll be brilliant. So now what is mistletoe? Have you seen this this kind of structure here in our campus? So basically all these references are relevant to US, Europe, right? So because we did not write plant pathology book, we only worship and leave a fate to the God, right? We don't study the reason, we don't look for the treatment, we don't do. If we get a smallpox, instead of going to temple, we go to, instead of going to doctor, we go to temple, right? So that is the reason that we still lag behind in science and knowledge. But these people are not like that. So they look for a reason. So I'll tell you. So across the globe, everybody was like this only. So whenever there is a mishap happens, any disease affects your crop or it affects you, they thought that it was the action of God. Not only us, even the Europe, Westerners, okay? But they changed that it was not the act of God, it was the act of causal agents like bacteria and fungi. So that we will see. Yeah. So this is not a pathogen, this is a plant. This is another plant. So it used to grow on big trees, like, right? And mainly there are, it's a parasitic plant. We also have cuscuta. Have you seen the cuscuta? The yellow one running? Yeah, it is similar to that cuscuta. There are different types of mistletoe. So this was the first one recognized as plant pathogen because in the US and Europe, they depend on conifer plants, conifer trees for wood, right? So whenever this mistletoe infects and grows on conifer, it destroys its cultivation, its growth. So they ran out of this wood. So they considered and recognized this as a first pathogen. There are few types: leafy mistletoe and dwarf mistletoe. Yeah, this is the life cycle of that mistletoe. So See, they've studied everything, how it infects, how it grows. So unless we know each and every step in the plant pathogen infection process, we cannot prevent that. Right? So unlike us, like we leave it to God that please save us from this mishappen. They studied each and every step. So this is seed from mistletoe. So it lands on any branch of tree, and then it germinates, and then it grows. So you see that this root transverse section, cross section. This is the epidermal layer. These are vascular bundles. So it uses mainly haustoria. So like us, like we have mouth to eat, right? So fungi. So they have it's a feeding mouth part. So this is not common across all the pathogens, but some pathogens they have this haustoria structure. So they will attach to the xylem vessel, and then they will take all the water, and it will attach to the phloem. So because phloem leaves, they synthesize carbon, right, and all the metabolites, sugar and everything. So they will transport from leaf to root. So if the fungi they have a connection with the phloem, it can take up all the carbon, nitrogen, everything. So basically, they also want to grow, so they infect the plants, right? Yeah. So basically, the slide shows the entire life cycle of the mistletoe on conifers. So for this disease, they also found a treatment. So it was very simple. Wherever you see the infected branches, you just prune them. That's it. So unlike fungi, this is totally parasitic, obligate parasite. It cannot grow alone. Yes, it can. Yeah. Custard also can synthesize photos, can fix carbon, but it cannot. Yes, this is obligate parasite. Yes, it cannot grow alone on its own like Cuscuta, but they have the ability to fix carbon through photosynthesis. Yes. So what? See, they can fix carbon, but they need nitrogen and other components, right? For which they need one host plant, including Cuscuta. That's why Cuscuta appear green in color. So the reason that we see green on leaf is that the presence of chlorophyll. The chlorophyll absorbs light in certain wavelength and emits the light in certain wavelength. So we are observing nothing but the emitted wavelength. So that wavelength is close to the green spectrum. That's why we see green. So wherever you see green, it means there is a chlorophyll. So in case of Cuscuta, mistletoe, and any plant. If you see, I think now we have started eating sprouted seeds, right? If you soak the seeds and then bind them into any cloth, you will see not a greenish shoot. You will see pale, right? Because it hasn't developed chlorophyll yet. And also you will see root. Root will be in white color because they don't have chlorophyll. But they do absorb light, emit light. But since there is no chlorophyll, we don't see it as a green color. Yes. So wherever you see green, they can fix photosynthesis. Yeah. The simple solution is that so wherever you see a growth of mistletoe, you just prune them. So that was the first treatment to the first pathogen. So in the history of plant pathology and disease prevention. Yeah, so this is another very dangerous ergotism. So the one difference is that usually we focus more on bacterial disease, a communicable disease, right, like COVID, right? So one person get infected, for sure another person will get infected if they came in contact with that infected person, right? But in case of this disease, ergotism, there was no connection between people. Whoever ate this fungal infected wheat, they got the infection, right? So they were really surprised that people are getting, so these are the symptoms of this ergotism. It's called several names. Another one is holy fire. It gives you like real abdominal pain, like food poisoning. So whenever you get a food poison, you get pain, right? It's nothing but a toxin, mycotoxin, that produced by the fungi. So you'll get a skin blistering and very severe lower abdominal pain, and yeah, hallucinations, and you go insane, right? And occasionally death. So one unique thing is that, as I mentioned just now, so in case of communicable diseases like virus and bacteria, so it tends to spread from people to people. But this was very strange to people. Even scientists, they could not figure out why the people were facing and experiencing this kind of symptoms. Then they studied, and then they found that whoever ate the wheat seeds affected by this fungi, Claviceps purpurea, so they were the only one who were getting these symptoms. So this symptom has not only caused this pathogen not only caused disease in humans but also in animals, because we eat the wheat and rye. They also act as a fodder, animal fodder, right? Yeah. So the disease was first identified or prevalent in the French. So they gave the name ergotism. Ergot means spur, like the pain that we get in the lower abdominal pain. And another most important disease is, is iris famine. It's caused by Phytophthora infestans. Okay. So it causes potato blight. So they are really, I read, have you read Agrios plant pathology book, Plant Pathology by Agrios? Anybody plant pathologist? No? Yeah, so there's a book, Plant Pathology. You can refer to your students if, by Agrios. So this is a Bible for us, for a plant pathologist. So almost all the material that I show here are from that book only. That's a very reliable source. So Irish and almost whole world, we were cultivating only wheat and rye and rice in case of Southeast Asia. So nobody was cultivating potato. Okay. So to the Ireland, like in 16th century, the US, they transported potato and made this Ireland people to cultivate potato. Okay. But in 16th century itself, people have started noticing foliar diseases, right, and above ground plant diseases. So there were diseases and there were less production of wheat and rye. Okay. So when they came to this potato plant from the US, the Ireland farmers, they were really hesitant to adapt this potato crop, okay, because it was new to them, right? So and it's exotic, and they don't know how to cultivate it. But the potato had an advantage, because you plant potato, you get huge amount of production yield, unlike rice, and you can survive on potato for longer time than the rice and wheat and rye. And potato grows underground. So they also had another advantage, that we see only foliar diseases affecting only the above ground plants. So potato grows below ground, so it will be advantageous to us. So it should be good for us. So they adapted potato cultivation in Ireland. Okay, so it's not a native to Ireland; it was native to US. So for 200 years it went fine. They cultivated it, and then they harvested it, and then they used to store this potato in the soil itself. They were not storing in the air on the outside. They used to store inside the soil only. So, so whenever they wanted, they dig out this ground, then take out the potato, and then they eat. That's how it goes. Why? Because why they were storing this potato underground? There were a lot of war at these times in 18th century, 19th century, right, between Ireland and England. So the British people, they come and then they steal this food from Ireland people. So they were storing them underground, right? So it was going fine for 200 years. After 200 years, one fine year, they started cultivating this potato, right? And they were expecting the potato, they grow nicely, and then they were expecting very high yield. Unfortunately, so as I mentioned, this favorable condition for a disease to cause maximum disease severity. During that season, it was too winter, it was too humid. There were like 100% humidity was maintained for a long time period. So this Phytophthora needs humid condition and cold condition for its growth. So it doesn't mean that there were no fungal pathogen before, but it needed the perfect favorable condition, optimal condition to infect more and to become severe pathogen disease. So one fine year that condition occurred. So all the plants across Ireland were infected by the blight disease, this Phytophthora infestans. Right? You see here there is one plant and the complete plant, was dead across all the Europe, and not only the plant but also the tubers, the potato, they had the infected blight symptoms. You see, this is how it appears. So you see the dark region, so they are nothing but fungi, and they contain toxin, right? It's called a mycotoxin, like cry protein, BT, brinjal, BT cotton. So that's a bacterial toxin, because the bacteria produces the toxin, right? Likewise, so the toxin from fungi is called mycotoxin. Okay. Now their property, everything is identified. So yeah. So if you eat this mycotoxin, you will get abdominal pain, you will get all the severe disease symptoms from the potato. See this picture. It's a sketch actually. At the time, maybe the camera wasn't available. So the sketch shows that a lady is like digging the ground to get the potato that was stored the previous season. So not all the plants are dead. Some plants are okay, and even the dead plants had potato. So what they did is they dug out all the potato, and then they selectively selected the good ones, which didn't have any symptoms, and then they stored them. So next season they were digging out, and then they found all the potatoes were rotten, because so here you see the discoloration, brown discoloration. Here you don't see. It doesn't mean that there is no fungus here. Okay, so this is another take-home message that you can take. Suppose if you see a apple, you see apple or potato or banana, right? If you see infection, especially fungal infection here, okay, you might see some growth here, some brown spot or something. Don't think that only the fungus is present only at this local region. Okay, the fungus will grow. It will be there. It will grow here. Okay, so wherever you see the dark spot or fungal growth, that's an advanced stage in the plant pathogen infection. Okay, in the growth cycle. But doesn't mean that there is no fungus here. Okay, so what fungus does is it infects I should have put that image. So I worked with the roots, so I'll tell you about the roots. So roots will have epidermis, endodermis, then cortical cells, right? And then you will have vascular bundles, right? And then pith region, right? So initially what it will do is it will land on the epidermis, and then it will slowly grow like through cortical regions. Okay, so it will occupy entire cortical region. Okay, so wherever there is a enough amount of mycelia, there only you will see that symptoms like brown or dark, some fungal growth here. But wherever you see a less number of mycelia, you will not see any visible symptoms. But doesn't mean that there is no fungi. Okay, so in case of chickpea or any pulse and any pathogen, so they have to come in contact with xylem and phloem, because that is where water and nutrients are available. So in cortical cells, they cannot get more nutrients. They cannot sustain. Okay? So they have to cross this endodermis to access this xylem and phloem, so that they can. So this Phytophthora is one of them. It also causes blight, but it reaches up to vascular bundle to get all the nutrients, right? And it's a rot pathogen. So whenever it reaches this maximum amount of mycelial growth in the plant, then it will start producing a lot of mycotoxin, and it will kill entire plant. Okay. Yeah, you see here you see discoloration because maybe here too many amount of this mycelia. Here there are mycelia, but it to grow to that maximum amount of fungal. So whenever you see some kind of infected apple, I would suggest you please discard them. You eat, don't give it to kids, okay, because they will get mycotoxin infections. Yeah, again, so we don't leave it to the God. We study what happens, what is the causal agent, right, and what is its infection pattern progress, right. So they studied, they identified. So these symptoms were visible. Okay. At the time also there are a lot of scientists, professors working in the Ireland, in the UK, but what they said is, so this symptom you can see whenever you soak or when you water too much amount of water to any pot, you will get this kind of symptoms. The plant goes soggy, right? Rice can grow with a lot of water, but if you grow chickpea in a lot of water, it will become saggy, right? It will become rotten. It's merely due to more water, not because of pathogen. So at the time, the scientists, they thought that this is due to more water soaking. So the plant took a lot of water up and then become soaked, and then you see the fungal growth. Because I mentioned, it was highly favorable condition. There was too much winter, too humid, too cold. So the scientists misunderstood that this symptom is not due to the pathogen, but due to the plants took up a lot of water. So that delayed another 5 to 10 years to identify the cure. So later only I forgot the scientist name. Sorry, I forgot the scientist name. So he identified that fungi was the causal agent, and that caused all the infection and diseases. So, but people didn't believe him, because at the time nobody believes anyone but the God, right? So nobody believed him. So what he did is, he did an experiment, very simple. So these are the spores. He collected the spores, okay, and then he did 2 experiments. One is he grew one pot in one area, without dusting that spore. So in one of the area, one of the pots, he added the spore of Spidoptera. So repeatedly he was seeing only the plot where he dusted these spores, they were only getting infection. But have you heard this Louis Pasteur? But he said spontaneous, right? It's totally contradicting, right? If you don't have a pathogen, you won't get disease, right? But Louis Pasteur said spontaneous, totally contradict, right? So this two discovery came almost at the similar time, right? So Louis Pasteur was wrong, right? So he was proved wrong, okay, by this, because unless there is a microbe or spore in a place, you won't get any growth of particular microorganisms, right? So this experiment, this Irish famine, it opened up so many people's eyes that we need to study plant pathology, infection pattern, and causal agent, everything, in order to find a cure. Okay? So he repeatedly proved that wherever he inoculated with a spore, he got the disease, and people started believing that. Then onwards, that disproval of spontaneous Louis Pasteur's experiment. Afterwards, people started working on and the birth of plant pathology. So far we have seen importance. Now we will come to tea. So we take now tea and coffee, right? So in the early times, people were taking only coffee. There was no tea available. There was no tea. So, but the tea came into existence or into cultivation because the coffee cultivation was completely destroyed by a rust pathogen. Okay. Majorly, there are only two types of coffee. One is Arabica, another one is Robusta. Right? So Arabica, So Arabica we drink. So we cultivate in South Asia, Iran, all the Southeast Asia countries. We grow only the Arabica, and Arabica is tastier than the other one. Okay? Yeah. See, as I just mentioned before, high yielding varieties, tastier varieties tend to be susceptible to plant pathogen infection, right? So this Arabica was really tasty, and unfortunately they were infected by this big rust. The name of the pathogen is Hemililia vastatrix. Yeah, this was the pathogen. It causes rust. I have seen myself rust in chickpea cultivation in Kadapa. I don't have the picture, but I found one particular area where I see a lot of rust, like kungumam, that red color. You will see a lot of them. And the rust pathogens tend to be obligate, not parasite. They say obligate biotroph. So in case of pathogen, they call it biotroph. So so there are two things. One is biotroph, hemibiotroph, and necrotroph. So this is all the rust ones. You cannot grow this rust out of, if you don't have any host pathogen, you cannot propagate this pathogen. You cannot store them. It will be very difficult to infect again. So you always have to take out the infected leaf that we can store. So obligate biotroph, they cannot grow without the pathogen. Yeah, as I mentioned here. So this map is showing the map from 1952. The whole Asia, Southeast Asia, they were completely infected because we cultivate mainly this Arabica species, and in case of the US and West America, and they were cultivating only Robusta. So they were completely disease free, right? And we had a lot of slavery in our country due to colonization. So when all the plants were like affected, then they wanted to have a alternate drink, then which became tea. So the tea were resistant to all these pathogens. So that is how we the tea came into popular cultivated crop, and then we started drinking both and tea and coffee. And recently there's a report from European some union government. They said that there is no neither nor positive or negative effect of drinking green tea. You take green tea, people say what's that? Antioxidant, right? No, there is nothing like that. Okay, so it's all lies. So it's a Europe Union some food some entity. So what they do is their job is to look at all the food habits of across the world, and then they evaluate like which one is good, which one gives nitrogen, which one is good this omega-3, omega-6. They are the one who found everything. Then they regulate the food intake of the whole world. So recently they released a document that taking green tea neither benefits you nor harms you. Okay. So, but it's good to take coffee. Coffee is good in a particular amount. I don't know. I can't tell you right now. I don't know how much it is, but coffee is good. Coffee we can take, especially black coffee. So that's the reason that in the US they have only the black coffee, the American coffee. So they take only water and the black coffee. Yeah, that is good for health. Yeah. So far we saw one pathogen, one plant, right? So it's not the case. So plants they coexist with more than one pathogen, like heat stress, drought stress, salinity, everything they grow like along. So you see here this plant, this is called Arabidopsis thaliana. Have you heard this term, Arabidopsis thaliana? No? Yes. So we have model plant and crop plants, right? So in case of so mouse is a model, right? Mouse is a model for our clinical trials, right? So we can't be the models, right? Likewise, in case of plant, we do almost all the work, molecular biology, in this plant, Arabidopsis thaliana. So it is not native to India, but it is native to Europe. And the reason that we selected, that we adapted Arabidopsis thaliana as a model plant is that its life cycle is very short. I think 120 days it will give you seed. Suppose if you genetically modify its genome, or you express any gene, or you silence any gene, so we need the seed. We want to see the effect on seeds, right? So that we can identify the mechanism, right? So this is very simple, and this belongs to mustard family. All your mustards, so they are in the same family. So this one's. So here I show that there are not only one pathogen, but a plant can be met with more than one pathogen. So here the first one is this is a healthy one. This is a gray mold. This is mildew plants, and this bacterial speck. Yesterday you might have seen a bacterial speck in potato, right? Yeah. So the Pseudomonas, okay, no, yeah, not here, maybe. And this powdery mildew. So all these symptoms are caused by different pathogens, different fungi and bacteria. Yeah, so I mentioned Haustoria. Remember, see the small structure here, like a bulging structure. So that feeds on xylem and phloem. So that extracts all the water and nutrients from your plants. Yeah. So there is always, whenever you see a susceptible plant, sensitive plant, you will also see resistant plant, because we have through cultivation breeding, we have a lot of cultivars, lot of breed varieties. So not all of them are susceptible, some of them are resistant as well. So that is where the breeders. So until recently, so right now we use CRISPR gene editing technology to develop resistant variety, right? So before the CRISPR was born, people were using the breeding tool. So let's explain in a minute. Yeah, you see here, just this leaf is susceptible because it has all the pathogen rust. Look at this one. This is not infected, not susceptible. It's resistant. So you see the yellow spot here. Here the pathogens landed, infected, but they could not, yes, grow further, and they could not make spores. So unless you make spore, you cannot, like they say, secondary infection. So the primary infection will start will start with first spore, then it will grow, then it will produce more spore, then this spore, the daughter spore, will go and infect other plants. It's called secondary infection. So if you stop at the secondary infection, still you can save the plant. So this leaf is showing resistant phenotype to this particular pathogen. Yeah, you see here another one. This is wheat. You see the first one is resistant, and there are resistant in susceptibility. It's not a black and white. It's not a black and okay, yes or no. So it's always quantitative. Like, so the genes which provide resistance, they are quantitative. So more number of that protein expression, more resistance, right? So you see here there's a gradation. Completely resistant, then less resistant, and less, and then this is completely susceptible. So for the potato, for the tea, all the resistant cultivars were developed through breeding. So the breeding, so I'm not a breeder, I'm a molecular biologist. So now we assist breeding. We help breeding using molecular techniques. So you might have heard the terms molecular assisted breeding. Yeah, so it's nothing but we study the gene, its function, everything, and then we tell the breeders, okay, this gene is present in this cultivar, and this gene provides resistance. Now what they will do, they will cross two different cultivars, like extreme susceptible and extreme resistant, of course considering their yield potential. So the yield potential is always main factor apart from this disease resistance. So they cross like extreme cultivars, and then they get a seed, and then they look for this disease resistance as well as high yield, right? Yeah. So I don't have a slide. So when we look at these breeders and their layout, it will look awesome. It will like they will have a hundreds of plots. So the plots will show you like more disease from one plot, and no disease one plot, and less disease one plot. It will look like a rainbow. So if I don't know if I have a picture. Yeah, basically the breeders, they save the world. So they bring I cannot connect my laptop. So yeah. Yeah, can you see here? Yeah. So all the molecular breeders in ICRISAT, so we are microbiology, so we don't do this, but ICRISAT they do this work. So whenever they want to identify a resistant variety, so they cross two extreme varieties, and then they get the seeds, because not all the seeds will be resistant or susceptible. There will be a lot of mixture because of its quantitative trait. And you see here, uh, these are completely dead and these are green. Also, we keep controls. We also cultivate known resistant variety and known susceptible variety for the control, that okay, this infection is indeed happen. So those things. So this is quite common. So I just wanted to show you this picture. See, all these play a big role, because before we use artificial intelligence or machine learning to train that model algorithm, we need to know everything about the disease, all the factors, favorable conditions, so that we can train better and then we can predict in the future. Okay? It's like a prediction of rainfall. Yeah, so the plants, so we avoid this disease occurrence. Either we can avoid them by growing plants in a different area where there was no disease was reported in the previous season, or we can exclude some of the pathogen, like we can apply chemical agents or soil fumigation to kill all the pathogens in the soil, or we can eradicate, like whenever the plant is dead, infected, we can remove the particular plant and then burn them up. So that also is possible. The last one is the protection. So here comes that molecular biology. So we study its infection pattern, and we study the resistant protein or enzymes or factors, and then we overexpress them, produce more in one plant, and then we see their response. All those things we do. Just a simple image from one of the plant physiology book. Here, this is the leaf. Maybe the botany people can relate. So this fungi, they can land over the leaf, okay, and then so we cannot eat wood, right? We can eat rice, but we cannot eat wood. You know why? Because we don't have the enzyme which can digest wood, right? Yes, like cellulase, right? Likewise, we don't have it, but this fungi and bacteria, they have the enzyme, cellulase enzyme. So what they do is they land on epidermis and then secrete the enzyme cellulase, and then they degrade the cell, plant cells, and then they enter through. And you see this growth of this fungal mycelia here. And have you seen stomata in plant? Okay, yeah. So wherever there's an opening, that way also the fungus and bacteria can enter. So whenever plant detects presence of bacteria or fungi, it will close the stomata. Okay. So what pathogen did? It secreted a protein. It's a funny story actually. So it's always who wins first. It's like a tug of war. So both pathogen and plant, they'll pull each other. So they'll keep winning. One will be losing, one will be winning, and another one will be winning, and another will be losing. It's like that. So there is one protein called quarantine. I think it's quarantine. So this is the maybe it's coronatine. So whenever the plant detects presence of particular pathogen, so it will close this stomata. Okay. So what pathogen did? It produced this molecule, and then it cheated plant. So whenever the fungi produces this molecule, the plant will think that there is no pathogen, and it will open up the stomata. So the bacteria will enter and then cause disease. So each bacteria, fungi, they have different kind of such protein factors, to overcome this plant defense. Yeah. Yeah, so treatments. So first and foremost is breeding. Breeding is the first tool that we can with which we can develop a resistant variety. Like that's a permanent solution. These are like temporary solutions: applying fungicide, soil fumigation, solarization, removal of infected plants, and intercropping. So this intercropping, do you know how it helps preventing disease? Any idea? Exactly, the sir is correct. So I'm going to give little like technical term for that. So we have a term host plant and non-host plant. Yeah. So host and non-host. So not all the pathogen can cause disease in all the plants. Okay. A particular pathogen has its own plant. Only in that plant it can cause disease. So if there's a pathogen, because it's an evolution. So host and pathogen, they've been evolving for a long time. They compete with each other. One put the plant defense and then one breaches the defense. So it goes like that. So particular fungi or bacteria, they can only infect very specific number of host plants. So in the intercropping, what happens is, suppose we grow chickpea and corn, right? Chickpea and corn. So the pathogens which can infect chickpea cannot infect corn. So that is how you can lower the secondary infection, right? And also you can increase the distance between two host plant like chickpea and chickpea, and you can reduce the amount of inoculum that it will spread to a next plant. Yeah, that is how this intercropping helps. So this is relevant to plant disease, but in case of fertilization, so legume plants, they fix nitrogen, right? So the studies are going on, and they also say that hypothesis that the maize and rice, they get the nitrogen from the legume plants. So that is how also intercropping helps in increasing the yield as well as protecting the pathogen disease. Yeah. So here comes, so once we know causal agent, symptoms, the factors favorable to the plant pathogen, now our job is to predict the occurrence of new disease or the same disease in the future. So that is where we use artificial intelligence. Right? So basically we predict in order to prevent the future disease occurrence. Yeah. So it's I was worried that yesterday was a practical and today was theory, because theory has to go first, right? But it's better now. So now you know how the machine learning works, right? So basically you collect all the data pertaining to your disease or whatever, then you feed into an algorithm and train them with the similar images, and then you take sample images or new images, and then you can see whether it's identifying or not. Similarly, so basically what we do, we teach that particular algorithm model, right, to learn the pattern, because suppose the leaf, like if wherever there's a virus infection, you will see yellow color, right? Whenever there's a bacterial infection, you will see spot. There are different spot, bacterial spot, fungal spot also is there, right? Sometimes you will see like complete drying of one side area, right? So we basically train this algorithm to learn the color, shape, the size, and different stages, right? So it doesn't do anything, it doesn't have any brain. What it will do, only it will know, okay, you are feeding a leaf with yellow color, that means this is virus, right? It cannot differentiate, it cannot think on its own, it will just learn the pattern and make decision. That's it. So basically machine learning, we are just feeding our own data, making it learning, and yeah. So yeah, so there are two paradigms, fundamental paradigms that in present in machine learning. Is it working? Yeah, so one is supervised, another one is unsupervised. So supervised is in our presence, like unsupervised is not in our presence. So whenever you heard artificial intelligence, it's called unsupervised. Okay, we train the model and then ask the model to take its own decision. But in case of supervised learning, we'll feed it all the data and then we'll give a known sample and we'll make sure that it is predicting correctly. But in case of unsupervised, it's otherwise. Right? Yeah. Basically, there are two paradigms, two examples, two types. One is supervised, one is unsupervised. Yeah. So in case of supervised, it's like a teacher. Like we show apple and then ask the kid to say it's apple, right? And also flashcard. Like we show animals, letters. It's like a supervised. Yeah. Likewise. So what the machine will learn? It will learn its color, its shape, its leaf, and its stem. So everything it will learn. So when we feed the image, yeah, it's just like teaching a child, this supervised learning. But in case of unsupervised learning, right? So first level we teach kid. The next level we give them toys and let them like this. So we'll ask the children, okay, you can segregate different types of toys, different shape. You can categorize them. So that is nothing but unsupervised learning. So we don't tell what toy is it, what shape it is, how many holes it has. We don't say, but that machine learning will learn on its own and it will predict. Yeah. So yeah, the unsupervised learnings will group different shape, size, everything. Just like you see here. Yeah, in case of supervised learning, it's not working. Yeah, so we give input, our images, features, whatever, and then we get the targeted outputs. It's very simple that we'll make that particular algorithm to learn how to map from input to output. It's very simple. Okay, so and also it's from one destination to another destination. You give input and it give you output. That's it, with whatever you trained. But in case of unsupervised, it will take its own decision. Different shapes, different sizes, and different types. Yeah. Yeah. So basically, we have to like yesterday we saw potato blight, leaf spot. Like we have to classify them, categorize each image, each category. Then we have to train that particular model in case of supervised. Okay? So it's like that spam email. If that email ID doesn't have your university domain, it will automatically consider the spam. So that is nothing but training only. So the people who have developed that particular software, they might have trained the software with spam and non-spam categories, images, inputs, and the tumor, malignant and benign tumor. Yes, it's like classified pictures and categories. So you train the model, right? So we need to identify, we need to make sure that whatever the trained output is correct. So there are some statistical models. One is regression. Right? So have you heard the linear regression, linear, multilinear regression? I think yesterday I showed my paper, right, where I have shown actual prediction, right, and predicted disease incidence, right? Yeah. So we use this statistics to see whether predicted result was correct. Yeah. So so this particular AA not only can be used in plant disease or yield, it can be used for anything, right? Anything for house, whatever. Like in case of stock market, they predict, right? In 20 years, this is how it will be, the price range. So they also use similar algorithm. So they feed the data for the last 10 years, and then it will ask the algorithm to predict how it will behave in 10 years, upcoming 10 years. Yeah. So this is the workflow. I think you are well aware that we have to collect the, you collect the data, categorize them, right? Like different types of pathogen, different types of disease, and then and then you train the data set, right? And then you validate the data set with the known image. You take your known image and then feed into the model and then validate it, whether it is predicting correctly or wrong. Yes. Yeah, so I forgot to mention. So it's nothing, so we need to train the program. Okay, so a lot of process like collection, preparation, training, validation, and then different type of model selection. So some model, there are some artificial neural network, convolutional neural network, computer vision. There are several models. Okay, so not all model will be perfect for all the application. There are some model will be perfect for applications. So we used artificial neural network for disease prediction. Maybe so likewise you need to train several models and select one of them, one of the best model for the future use. And then you evaluate the model using the regression statistics, and then you test with your unlabeled data, means images from the field. So what do? We get the data, we label the data, label different disease wise, categorize them, then we train the model, okay, and then we validate with known images, right? Once you have done everything and you think that the model is ready, you take the model and go to field where you get the images, again feed, and then you test. That will be the final test in case of supervised. So of course there are a lot of challenges, right? Yesterday some people said that they gave only 4 images as an input, and then they predicted that both categories were predicting like 80% or 20%. So it's called underfitting. So you need to train the model with too many images. So yeah, these are like just a technical term with evaluation, overfitting, underfitting, the quality and quantity. So the quality, the picture has to be really in a high quality picture, right? And the quantity, number of images that you feed. So more the number of images that you feed, better the accuracy. So underfitting is that. So there was a download option, right? There was codes that we downloaded. In that, you can change the code. You can change that this model needs only 100 images. That is more than enough for its prediction. Some models need only 10 images. Some model may need 1000 images. So those changes you can make in your model. So the model which takes only less number of images, they are very simple model, right? You cannot rely on them more. So this is called underfitting. So overfitting is that sometimes it will predict wrongly. Sometimes when you between fungal spot and bacterial spot, it cannot differentiate. So we call it like outlier and noise, right? But all these things can be rectified if you use clear images and more number of images, quality and then quantity, with which you can overcome all the limitations with this one. Yeah, let's see unsupervised learning. Yeah. Okay, here this algorithm is totally different. Okay, if you ask me, I have no idea. Okay, I'm a molecular biologist. I'm not a systems biology. Maybe Vivek sir will be coming. Vivek, he is a professor in systems biology, so he must be able to help you. Okay, so yeah. So in my research, I used supervised learning only. So we fed all the data, and then we asked it to predict. This unsupervised is for the professionals in systems biology. So I don't know what kind of algorithm they use. So basically here, we don't categorize them like different disease types. We don't do that, and we don't label them. And then, but we use the same data to train the model. So what it will do is, without giving the disease name, it will just give you images with a similar phenotype, similar symptom, similar leaf area. It will give you. So this is very like useful, when you have a very big data, like maybe millions of images. In such cases, this unsupervised learning is very useful. For a small scale, for one pathogen or one crop, maybe 10 pathogens, the supervised learning is, that algorithm is more useful than this unsupervised. Yeah. So here when you input your features, so yeah, so the main goal of this to is find hidden structures and pattern and groupings. What can be the examples here? Yeah. So the clustering is, yesterday we saw, right, the scatter plot. In the scatter plot, you fed, then it gave you different sizes, right? That's clustering. So based on the category, it will give you some pattern. No, I mean like huge number of data. Where will they use? Maybe in case of climate change prediction, maybe they may be using this unsupervised learning algorithm to predict whether the future weather changes or climate changes. Yeah, so like he pointed out, so there are 3 components to unsupervised. One is clustering, another one is association, another one is dimension detection. So have you heard this principal component analysis tool? Principal component analysis tool. I'll show you image. Yeah, this one. It's nothing but your scatter plot. It's advanced version of scatter plot. So you will have 2 different dimension here, like basically 3 dimensions, or people go for 2 dimensions. So it will segregate the data, cluster the data based on 2 different categories. So maybe we'll see some practical. I will explain you in chalk and board. Was? Which one? No, that will also cluster. No, we use regression to see the actual and predicted one, and also we use PCA to see how this algorithm like differentiated different components in the data. So we use this when we want to see a different dimensions in a particular data set, we use PCA. So I will explain. Yeah. So basically, the first one is clustering. Clustering is nothing but like a classification of different groups. So the model itself will classify. Unlike this supervised, where we label class 1, we label, right? Class 2, likewise, we don't label. So when you feed like 1000 images, it will classify itself, and then it will give you pattern. Okay, out of 1000 images, 500 are like this, 200 are like this, like that. It will classify. It will give you cluster 1, cluster 2, cluster 3, cluster 4, cluster 5. Yeah, likewise, it will give. Later you can, after it clustered, you can label them. Okay, but the initial work will be done by this one. Because see, for example, if you go to a field, you take 1000 images. So in the camera, every time you cannot change the name of the image, right? Usually you click one image and then change the name. Click one image, change the name, right? But that's not possible. I don't know here, but in the US, the US geographical area is 2 times bigger than India, right? So and they grow only soybean and corn. They don't grow any other plants. They don't grow rice. They don't grow wheat. No, they grow only 2 crops in an area which is 2 times larger than India. So in that case, if they want to study some disease incidents or pattern, they can't just go and take one picture, label them, right? So what they will do, they will take drone and they will take like thousands, millions of images, and then they will ask the AI to categorize them. So once the AI categorized all the images into different category, then they label. Okay, this is this soybean plant, this is maize corn plant, and this is this infection, that infection. They will label. Okay. That will do basically clustering and classification of millions of images. Yeah, the segmentation and classification, they are all like similar terms. And association. So association is nothing but labeling. So you categorized, so you asked this algorithm to categorize, right? And then you have to associate a particular category to particular disease or particular crop, right? Now, so far there is no labeling, right? So, but it categorized millions of images. Now you have to associate different category to different names. So that is called association. So you so the variables are nothing but categories. You get the variables, and you like these are different examples. Like here we only mention plant disease and crops, right? So and then yeah, and then so far you have no idea what data it is, what is there, no idea, right? Because there are millions of images. Now you want to look at all the images in a simpler term, right? In simpler term, like this PCA is very this principal component analysis. It's a very effective tool in molecular biology. People who are working with the transcriptomics, proteomics, metabolomics. So whenever we do transcriptomics, we get thousands of upregulated genes, downregulated genes, right? So we cannot go to each and every single gene and then see how much expression, how much we cannot see, because there will be thousands of genes, right? So if you want to see them in a simpler form, this PCA is the best one. So PCA usually there will be a y-axis, x-axis. We say component 1 and component 2, right? For example, component 1, you say height, and you say leaf shape, okay, from narrow to broad. So the principal component analysis is itself an algorithm. It will make its own pattern, like unsupervised. Okay, you feed the data, it will take up its own amount of expression, size, everything it will take. So if you take height, maybe you can short and tall, and you have a data from corn and soybean. Okay, so if you want to see maybe amount of area of cultivation, so what it will do is, suppose if you have taken like 100 images, if it is maize, you will see all the like narrow leaf, tall plant, you will see all the spot here. And if the images are taken in soybean, which is a short plant with the broad leaf, you will see all the point here. Okay, and if you see some other weed, with a like leaf size, leaf shape in the mid range, you will see somewhere here. So basically, this will give you like proper clustering of huge data. So with this data, you can easily identify, okay, there are many like maize plants, there are many soybean plants, there are some intermediates. So likewise, so yesterday we saw that software, right, orange. So if you want to know what are these points, you can select them and then it will give you a separate list. So that's the use of this component list. So usually it comes like we can like mark them with a round. So when you have a large data, huge data, maybe in the students, if we have we have NEP program. So we get in one class 90 students. So the 90 students, you have a mark, and if you want to see who got more, who got less, we can use this principal analysis. It will tell you. So instead of looking at the 100 row in the Excel sheet, just you feed the data into this principal component analysis tool, like software, statistical software. It's going to give you some category. So this many people are doing well, this many people are not doing well. So basically it will give you a dimension and it will simplify large data. So we use this to validate this unsupervised learning algorithm. Yeah, you see here. So this and this are related, right? This and this, but this and this are far related. So here you see three dimensions, but we usually go with the 2 dimensions, just x and y. So it will give you. So depending upon your data complexity, you can increase the number of dimensions, number of components. Yeah, so association, again the same association. Oh, maybe I'm going upstream. Yeah, so this is pretty much about artificial intelligence and its usage in predicting disease model or any fertilization, anything. So basically, you need a data set to train your model. There are several models. You have to pick one model, simple model, train the model, and then validate the model, and then you test the model with the real-time images from the field. So these things we already seen yesterday, how the disease causes yield loss, right? Yeah. Yeah, this is very easy. Maybe you can teach your students, right? So teachable machine, it's freely available one. Yeah, very simple. And this software, this app, you can suggest to your maybe village people, they can get benefit from. Yeah. So this is for our faculties. For faculties and researchers, this is also freely available software you can use. Yeah, I think pretty much this is it. I can go deeper also. Yeah. So if you have any questions, I can answer. So I have a request to you, all of you. You can. So I have started my new lab last year. So if you have any potential candidates, potential students who has a scientific desire, you can ask them to contact me for any position. So I have written some project research proposals. So if you get a research proposal, then I will have a vacancy for project assistant and project associate. So they will be paid 25,000 per month plus 30% HRA. So yeah, if you have a potential students, you can help. See, MSc. Yes, MSc students. After MSc, ma'am. Yes, ma'am. No, any life science, ma'am. Botany, biotech, microbiology. Yes, but not zoology because we work in plant science. So, botany, microbiology, biotechnology. You can suggest them to contact me. Sir, email ID is there. Email ID will do, I guess. And also, you can call us for invited talks. Of course, I'm going to share. I'm going to share the entire PPT. Yes, ma'am. I will give it to the Priyanka. Yes. Ask them to write to me. So projects, we will take if the students are willing to take 6 months project. 2 months it's less likely because for 6 months, yes. Yes. Sir. Sir. This one, sir. Vadivel. Ma'am, so MSc people has to do some project, right, for 6 months. They can come. So yes. So they can write to me, or if I don't have a position, I can refer other my colleagues. Okay? Yeah. So that is how everybody is known only. You apply for faculty position. Yeah. Yes, PH position. Yeah, you can apply. Yes, ma'am. Interview based. Yeah. It varies. There'll be interview definitely. Exam also can be there. Yeah. Yeah. If there are too many applicants, then we'll conduct exam. I think we are pretty much close.
Okay, so I presume just like me, you all are also teachers in teaching UG and PG students in different colleges. Okay, now yesterday's focus was more of on AI in the area of wildlife. Is that creating a problem, my mobile? Do I have a device to use for slide transition? Just check with the. Okay, sorry for the interruption. So, yeah, yesterday they talked about, yeah, so other than Manas, who all came? Can anyone tell me? Who? Vadivelu. Okay, what did he cover? Right, so means in pathology, how to use AI models. Yeah, in diseases, plant disease, okay. Not only plant disease, I think he might have also touched upon the taxonomy, identification of plant species and other. Huh? Siddharthan, he covered that, right? Yeah, good. So we'll, in the last 2 days, I think we'll slightly shift the focus and we'll talk more of the upcoming areas of biology, like biotechnology or applications of AI in diverse areas of biotechnology. Okay, so biotechnology itself is now diversifying. Not only biotechnology, we'll also kind of touch upon microbiology. So when I went through, I think there is focus on these two aspects, biotechnology and microbiology. Okay. So, yeah. So today what I'm going to cover is perfect. Okay, so as I said, I'm at School of Life Sciences in the Department of Systems and Computational Biology, and a couple of more faculty. They will interact with all of you for different themes and different topics. Okay, so moving on. So given the agenda, I think we were suggested that we should at least cover these two aspects, application of AI in genomics and also in protein structure modeling and drug discovery. And I'll give a brief idea about that, okay? So that the lab sessions can be relevant for you, okay? So I'll start with genomics followed by the other topic. But I mean, one thing that is interesting is that such sessions, given the change in technology, I think such sessions have become really very futile in the sense if you go and use any chatbot, okay, let's say Gemini or Copilot or anyone, or DeepSeek, and if you ask applications of AI in genomics, right, they'll give you a overview, they'll give you a lot of relevant information. If you want a detailed information, you have to prompt that chatbot to provide you the detailed information. If you want a review article, they'll give you review articles also, okay. So you can ask the chatbot that please guide me, please provide me relevant information in this area or in this area. So this means technology has changed, and I think you'd be also aware of these things that while you'd be teaching in the classroom, students are also on your mobile, they are also checking a lot of things, okay. A lot of things means let's say there is anything which is not clear to them, you use a terminology, okay, and they will quickly check. For example, if you say that you use a terminology Shannon entropy, they will immediately check. Although you are teaching ecology class, you assume that they understand Shannon diversity, but they can quickly check that. So like this it goes. Sorry? Checking and cheating, right? Yeah, so that's true, right? And we'll have to be mentally prepared for that. Okay, so our role will be getting diminished with time, and I think our role will be kind of a facilitator who just connects these two things. Even I don't know for how long this facilitator role will be there. I doubt, but currently this is the role. Okay, so things are changing pretty fast. Okay, so moving on. So before we delve into this area, okay, what are the key areas of genomics and where AI is contributing? If AI is contributing every aspect, not necessary. Those things which are really, which has been solved and for which cost-effective tools are available, then AI won't invest much resources into that. So we'll have to find out what are the areas. So the word genomics comes, I think while you may be faculty members of different discipline, but this is genomics is something that connects you all, whether you are a microbiologist, you are a virologist, you are a plant biologist or an animal biologist. So that theme connects you all, right? All of them, they have a genome and their phenotype, their genotype and a lot of properties, it is guided by the information packaged in the genome, right? So what are the key areas or emerging areas which are worth looking into, particularly from the perspective of this thing? And when you teach genomics, so do we have a faculty in genomics here? Anyone who teaches this particular course? Functional genomics or genomics? Sorry, you have? You have one paper, but are you teaching that? You are teaching that. Very good. So at least we have one faculty who teaches that. So what are the key areas? So the moment we say that genome, so one has to understand what are the characteristics of the genome, how big they are, and but that is based on the known information. We already have a lot of genomes available in different databases, whether human genome, corona genome, any genome you tell, and most of the genomes are already available. We have to just go to a relevant website, go and check the properties. Okay, whether it is linear or circular. Okay, if it is linear, how many chromosomes are there, what is the size, so on and so forth. That information is pretty much available, right? But beyond that, so where this area is expanding, this area is expanding in terms of genome sequencing and assembly. So while a lot of genomes are available, your genome is not available, my genome is not available, and I may be interested in that. Okay, or a population genome may not be available, right? So this means still, even human genome, thousands of human genomes are available, still money is being invested in this area of sequencing and assemble the genome back. Now when I say sequencing, so sequencing is a technology, you all must have taught your students Sanger sequencing. Okay, the one which is based on dideoxy ribonucleic acids. Okay, so or the various types and what is the protocol. So you might have taught all of them about the basic protocol, and the current technology sometimes uses similar protocol or sometimes uses very innovative ideas. Anyway, today's purpose is not to get into that, but genome sequencing and assembly is the area which is still being used, and it is practiced a lot. And once we have a genome, it is still relevant. Okay, variants. So while human genome is available, but we all differ from each other. There are 30-40 people sitting in this room. We all differ from each other in variety of traits. Okay, what are the alleles which correspond to that? Okay, we don't know which allele we are carrying. So this means variant prediction and annotation. Comparative genomics. Another thing is that not only we have a genome, we also often compare the genomes to learn from the existing genomes, right? So comparative genomics. One more aspect, functional genomics, very, very relevant area. The one which actually gives meaning to the genome, which characterizes what are the functional features associated with different regions of the genome, different locus of the genome, different alleles of the genome, right? So functional genomics is another area. So these are the areas which are expanding, but not necessarily genomics means only these areas. There may be many more areas like personalized medicine is there, right? And so on and so forth. There may be many areas, but we'll touch upon these areas. So current challenges in this particular area. So one topic I have picked, and then one by one we'll delve into that. So this image I have copied from ResearchGate. Okay. Anyone who is familiar with this image? Definitely yes. Anyone familiar with this one, this image? Or anyone who would like to explain this image? Yeah. So the data that you get from Sanger sequencing, right? So basically we get what? Chromatograms, and we get the called bases. Okay. So they say that, so this is just a small, for example, if you have sequenced amplicon of size 300, okay, so you will get a sequence of size 300, right? Or plus minus, right? But along with that, they give you this. What is this? These are not readings from BP machine. It's a chromatogram. What does that correspond to? Anyone? Wild guesses are fine, because not all of you are from the same field and you are teaching sequencing. So based on the context what he mentioned, what do you think? What is this? Yeah, observed? Absorbance. Okay, but that's fine. Anyone else who would like to refine his answer? So basically you can see that there are 4 color codes, right? Black, green, blue and red, right? And then you can see that there are some 4 waves, or which are depending upon, so this means, for example, if you track green, okay, so over here, let's say start from here, so the green is over here, means the amplitude of green is 0 here, then it peaks, then comes down, then it peaks, comes down, then again green is here. You can see that, or maybe not, if may not be very clear, but it is there, then it is still there, and then again there is a peak, and then there is a peak. Okay, and how does this peak translate into AA, and then again AA? Now you know what is the meaning of this. Okay, so this means the height of the peak tells that how reliably we have identified, sequence that particular base, which is called base calling, right? So this means you can see that it's not much confusing, but there are few cases, for example, this one and this one, where the intensity is not that good, but this is the ideal one that you are seeing. There can be bad ones as well. Okay, just like in movies we have a hero who will do all sorts of things, and there are some bad characters also. So that's how producers or the writers weave the story. Similarly, particularly from the ends, okay, the 5' end, 3' end, you may not find very good quality, the peaks having high amplitude. And what does that mean? Means if so there you may find that multiple colors are forming, so multiple peaks at the same time. So for example, over here it is, let's say, black. You will find that black is also there, maybe green is also here, but the intensity is varying. In that case, no one can tell that what is the actual base lying over there. Okay, so such bases are called, but their confidence is low. So they are called low confidence bases. And when you are sequencing a genome which has billions of bases, millions of bases, imagine how many times such things may occur. So this means this is a routine problem, right? And there have been, what has been the approach? What has been the current approach to solve it? So we'll come to that. I'll switch over to another topic, which is, so this is about sequencing, this is about assembly. If you see, they will so for example, this read that you see here, similarly the bioinformatics software, which will find that are these reads sufficiently overlapping, right? And if they are overlapping, you will come out with a consensus string or a sequence, and you call that, oh, this is the genome. I showed you a small region, but then in reality you will have longer reads, hundreds and thousands of reads. Based on overlapping reads, you will come out with a assembly. This is called assembly. So I used the word assembly. This is what I mean by assembly. Now what can spoil assembly? What can spoil a good assembly? So for example, over here looks like pretty good assembly, but what can spoil assembly? When we are assembling these individual reads coming from the machine, so this might have come from one reading This might have come from another reading, multiple fragments. Their data is being stitched. See the overlap. GC, this is sufficient overlap, and that's how we have stitched it. Okay. So, what can spoil assembly process? Gap. So, what possibly he means that, for example, we have this read, this read, this read, and what if these 4 reads are not there and only the last 2-3 reads are there? So, this means you come out with a region where there is no sufficient overlap. So, the confidence drops, right? Or there are repeats and others. So, this means genome assembly, ideally it looks like it is a simple process. It is a pretty complex process, and particularly assembly of a bacterial genome, viral genome is easy, smaller, very compact. Okay, but then assembly of longer genomes, okay, it is difficult. Okay, it's like if you are asked to if you are also delivering a dialogue in a play, let's say on a teacher's tray, you are invited to deliver a dialogue by your favorite actor. One dialogue is fine. Delivering the dialogue of entire movie, that may not be easy, right? So you can imagine the challenge. So this means challenges are there. This area is growing, frequently being used, but some challenges are there. Okay, and this is how currently being done. So this is a clone contig based approach, which traditionally used, which was practiced, and when all of us, I think when we were in high school or colleges, that time I think this was practiced. Currently, this is the current approach where you take a fragment, right? Whether you know the map or not, doesn't matter. You fragment, shear it into pieces, which is called shotgun. You assemble them. You may get multiple short assemblies. See, this came from this single sequence, but how many assembled products we have? 2, right? So this means assembly is not an easy process. And then finding an order, so far we used to use, so far genome maps were useful in finding the order of contigs, right? But not all the time genome map may be available, right? So this means this area has lot of challenges, right? Lot of challenges, and the sequencing companies or the researchers, they are trying to fix some of these. So what are those areas that they are trying to fix? Yes, sir. From the reads, so for example, multiple copies of this sequence was fragmented, sheared, and the smaller chunks were sent for sequencing, for example, like this. And these chunks, we had the data and we assembled it like this, and this one is this one. Yeah. Yeah, that's what I'm saying. So imagine this entire chunk was sequenced. After assembly, we got only 2 assemblies and some unassembled regions, right? And when we look at the markers, the genetic markers, okay, that we have studied in genetics. So D and E, so one fragment contains D and E, this region, right? And another fragment contains H, right? So this means now you see how much you are able to assemble it in reality. This region, and let's say assume that from this only these 2 regions have come up. So this means there are gaps in the assembly, and the gaps are only, you also mentioned that reason why gaps could be there. Could be because of repeats, okay, shallow sequencing, smaller reads. There are many factors. That is a topic of some other class. This one, this is not a sequence. This is a map. It's like, how did you come to this place? University of Hyderabad, this building, how did you come? You must have used a Google map or something to track it. So similarly, a genome map, it can be constructed based on genetic crossing, okay? And from the crossing, we know that what is the recombinant frequency of DE, how far. So this is called mapping the alleles and the morphological markers, genetic markers. So based on genetic map or physical map, we do have a map, but we don't have a sequence. Okay, so this is, anyway, if you want the details, you can go and look into genomes, which is a classical textbook for that. So these are the areas, gaps you have realized, and how. So for example, one of the companies, so there are many companies who are in the business of sequencing, and they generate a lot of data, something like this. You might have you heard of any company which is in the area of sequencing or next generation sequencing? Illumina is one. Anyone else? There are many companies these days. Okay, they all are competing. They come out with a very attractive package that we can sequence your microbial genomes, any plant genome, pathogen genome in this cost, this much data, so on and so forth. So this means that area is advancing. And for example, you mentioned Illumina. I went to the website of Illumina to find out how Illumina is trying to improve the problems that they are facing, right? And Illumina, one of the problem with Illumina is that their sequencing data is particularly fragmented. I mean, typically fragmented, shorter, and towards the 3' end, okay, they have a lot of low confidence bases. Okay, what is low confidence? I have already explained. Now they are using the power of AI to optimize the base calling, so that even if there are some poorly called bases, okay, based on the training models that they will train on their data. How they will train? What they will do? They will take millions of nucleotides, okay, so millions of nucleotides which were really poorly, I mean, for which the confidence was poor, and then they will check the actual, they will generate the real data. Means while their machine tells that not able to pinpoint what was the base, but through Sanger sequencing or any other technique, they will find out what was the actual base there, right? So they will find out that, then they will train that given this profile. So if for every such position, they will have profiles of A, T, C, G. So 4 chromatogram they will load for the fluorescence, whatever was detected by the sensor, okay, based on which they coded that whether it is green or blue or red or whatever it is. Same data they will feed to the model, and they will tell that when the profile is like this, the real base is this. Okay, so that way they will give millions of profiles. Okay, so for example, on this scale, they will give, okay, for example, this is a poorly called base. Okay, so here on the scale of 100, let's say this is 20, and then the green intensity of green is 18, the intensity of red is 15, and the intensity of, let's say, what is the other color? Black. Okay, it is 5. So they will train this, and they will tell that the actual base was actually G. So they will do the testing at their end. So they will take this data, train the model, and tell that when such a profile is there, 17, 15, 15 and 5, the actual base was actually this, right? This way their model learns that in which combinations it can predict correctly and which combinations it cannot predict correctly. Then wherever there is a poor quality, they will use that model, they will call that model. Can you, am I too loud? Sorry, where was I? Yeah, so based on the model, now they will apply it. So now you understand. So what is the take home message for teachers? So for take home, so while for us, there is not much scope to do, but then when we train our students, we can give an example that how AI, because our students in future, they will go and they will work on what are developing technologies, developing algorithms and so on. So they should know, so 2 things they can learn. They should know the gaps, right? And they should know that how such gaps are being addressed. And in future, if there is any other thing where you realize that there are gaps, I think a similar principle can be applied, right? So this is how they are taking care of base calling optimization. And a related thing is error correction. Wherever there is a sequencing error in general, okay, currently there are techniques, but now the data-driven technique is being used. So this means data-driven. So it doesn't mean that error correction in the sequencing errors are being corrected because of AI only. No, there were techniques and they were working very well. Okay, but there were limitations as well. Their accuracy was not 100%. And when AI based approaches are being used, if they find that yes, accuracy is better, they will prefer AI. So this means many algorithms are there and AI is competing with those algorithms, and in many cases AI is performing better than those algorithms. So this means there are two things which are competing. One, scientific principle based or algorithm based, computational algorithm based predictions, and this another is database. Not data science based, means data based, something which is observed and then that is trained, and that is, it doesn't require scientific principle. Okay, it just learns that what is correct, what is incorrect, and then it will do. So two different approaches are being used, and currently the purpose of this session is about how AI is being used to do that. Okay, some cases user, yeah. Yeah, so is there a way to do for specific thing, or yeah, so there are many papers how they use this deep learning and neural network to improve the base calling. In fact, Illumina would have published it. You go to the Illumina website and find out there are tools that I think they must have published the technology, how they have trained their model. That I can't assure that whether that is publicly available or not, but papers you may find how they have trained it. You may not find the actual package, you may or may not, but the methodology, how they have done it, implementation, those things you may find it. From Illumina, they have the link. You can go and explore it. Yeah, practical one I'm saying, how it is being done, what is the internal architecture. Okay, we will show you few more things. Don't worry, one or two specific cases we'll talk about. And then read assembly and acceleration. And I also talked about one issue, that to assemble a genome into bigger, the complete genome, that is a challenge. And again, over here, they are using AI models to do that. Anyway, I think given the time, we'll have to move on. There are many things to cover. Another aspect is genome annotation, right? And when we say genome annotation, means having a genome, let's label it, mark it, that where all genes are there, where all protein coding genes are there. In case there are protein coding genes, what are they encode for? What is their function? Okay. If at all there is a, we say that there is a gene, then what is the position from it starts? What is a okay, so what is the position from where it starts, so on and so forth. For example, I copied it from, I went to a database, a genome database, and I said that, can you display annotation? When I say, can you display annotation, what it has done, it has given the entire gene sequence, okay, and is there an issue with this? So it has given the entire gene sequence, and then it has labeled that from this position to that position, there is a 5' UTR, then first exon, exon boundaries, where are the exon boundaries, where are the intronic sequences, okay, where are the next exons, so on. So it has given the finer details of the gene model, right? So now that we have the finer details, that's what, so the purpose of annotation is to do that. And not only that, it will also tell that at a given locus, how many alternate splicing products are there. So that information can also be obtained from such sources, okay. So, but do we have how precise our annotation is? So if at all today someone's, for example, his genome or my genome is sequenced, for human genome we have pretty much good information and we can annotate it very accurately. But for newer genomes, okay, it is still challenging, okay. For those taxonomy groups in plants or in other areas, okay, which has not much explored, okay, then there may be some issues. And how they are solving this issue? Again, based on the training model. So this means they are using AI as a prediction tool. They will train them with existing information and then they say that can you predict it nicely, okay. So this is how they are using it. So moving on, so again there are a lot of gene prediction algorithms are there, or annotation pipelines are there. NCBI has a pipeline and many other portals have a pipeline, but AI is even improving that. I mean, accuracy of AI is even better than that. So this is another area where AI is being used to do that. But most important area where AI is used in genomics is the area of variant prediction and annotation. So this is this slide is from my classroom. Okay, I've simply I stole it from there. So what it shows, it shows that there is a reference genome, can be human reference genome or any reference genome, and these are, each line represents a read from sequencing data, right? And you can see that the highlighted region is nothing. This is a position where there is a variation. None of the other areas which are not labeled means there is no variation. So this means this reference is already available in the database. This data we got from sequencing, and when we align it, when we map it to the respective region, we find that there is one position where there are variations. Look at the variation precisely. So reference has a allele called A, and then in the sequence data you find that CAAA, CCA, CCA. So this means two things are occurring, C allele and A allele. A is same as the reference allele, right? And C is a new allele, which is in technical terms it is called reference allele and alternate allele. So two alleles are there. Now, so this means, so now we have two alleles. So we would like to know whether this is indeed there is a SNP here. Okay, that's a common question we ask. Okay, whether there is a SNP here or not. And many softwares are there. They will look at the quality, so many aspects, and they will tell that yes, there is a SNP with this confidence. That is, so this is called variant identification, and based on that, if there is a diploidy, we'll say that the individual which was sequenced must be diploid and it must be heterozygous at this position. Why? Because there are two alleles. And how, on what basis they are saying heterozygous? Because a frequency of both the alleles are equal. So this means that when the homologous chromosome was being sequenced, okay, so I think one chromosome had one allele, another chromosome had another allele at that position. So that's why we got set of reads supporting one allele, set of reads supporting another allele. So this means diploidy, heterozygosity, that is clear. But do you think that this pattern is always like this? No. In reality, you find a huge range. What you may find is that you may find a situation where C is occurring only 1 out of 10 reads and A is occurring 9. What will you infer? C, you find one read supporting a new allele called C out of 10, and remaining 9 reads are supporting A. Will you think that this is a sequencing error, that's why C is there? Or is it a genuine allele? That's where the software, whether bioinformatics based or machine learning based or AI based, they have to perform. So their power is in finding in true, that's called variant calling or variant identification. So they have, even there are complicated scenario, so they have to tell that whatever the variant they have identified, it is indeed true. Because it matters a lot, right? For example, someone has a rare disease. The clinician, the geneticist and the clinician, they talked and they say that we have not seen such symptoms. Let the exome I mean the exome means all the coding regions we sequenced. And then the bioinformatician tells that we have a position where there is a SNP. But then, first of all, they will ask how confident you are about the SNP. And then he has to tell it how confident he is based on the softwares. Softwares have to tell the confidence, right? So this means in such cases, imagine how important it is about, I mean the accuracy of the prediction, how important it is. Sometimes it becomes very important, right? Then he has to also tell that another word called annotation. If what is the biological relevance of this mutation? Can it impact? Can it cause any phenotypic change? And that also one has to predict. One cannot go back to the lab and no, we'll test it. We'll test it in mouse model, we'll test in this model, that model, not possible. And there are softwares which take care of this, right? So not only you have to tell that there is a variant and you have to report it to the clinician or the doctor at the patient. Also you'll have to tell that whether this mutation can cause any impact. So next comes impact, and you have to predict the impact also. But if all, so for example, this causes a change in the codon, but a lot of times there are synonymous codons, the meaning may not change. What if it causes it is a non-synonymous mutation? So SNP causes a change of the amino acid. So what is a non-synonymous? You must be familiar with that word. Non-synonymous means where the meaning of the, so not only the codon sequence changes, the amino acid that they encode, that also changes. So that's a non-synonymous mutation. But if all amino acid changes means it can affect the protein function. So he's, what's your name? Shrideep. So he's saying that if it is in the active site, it can affect it. Any other answer? Okay. Yeah, 3. We have 3 answers, kind of related answers. Any other answer? Anyone who disagrees with your colleagues or fellow trainees? Anyone who disagrees with them? For example, since we all belong to the teaching community, for example, you happen to be absent one fine day, right? And so what will happen? So let's say you are absent for a week. What will happen? What the HOD or the dean will do? Or if you are HOD yourself, what you will do? Huh? Class adjustment is there, right? So this means, so for example, if you are at Faculty of Life Sciences, what kind of adjustment your HOD may think of? They cannot go to psychology, they cannot go to sports, and they'll say that no, there are faculties, but he won't do that. Do you think that nature doesn't know these rules? Nature very much knows this rule. In nature, do you think that the protein sequences which is in your body, they are identical? No. There may be variations. Do you think that all the variations reflect into the phenotypic change? No. There are a lot of things for which the protein sequences may differ between you and me, but the phenotype doesn't change. In which cases? Is it true? Yes, it is true. If you look at the evolution, there is a tendency to, naturally, spontaneously mutation occur. And there are mutations which are called favored mutations. Okay. So this means even if there will be mutation, then the nature of protein, nature of function won't change. Okay. For example, glycine replaced by alanine. In nature, it is not a very radical change, but alanine replaced by proline or cysteine, it can dramatically affect the. So this means not always change in amino acid means there is a change in it is going to have high impact. So this means you will have to also see change in amino acid can do truncated mutations, right? What is a truncated mutation? Means insertion of a stop codon, and then the protein becomes meaningless, right? Not always all the time this happens. Most of the time happens change in the meaning, and not all the time change in meaning means there is a change in the phenotype, some alteration will happen. No, no, no. Most often alteration may not happen. You will have to check there are something called substitution models. Okay, one of the famous scientists called Day-4, she came out with a substitution matrices 50 years back, and she is the founder of, you can say, bioinformatics. The bioinformatics discipline was founded by her, and they used to look at a family of, so initially the protein sequences were synthesized, and they had data of lot of protein sequences. Okay, and based on that they used to see what is in evolution allowed and what is not allowed. Some mutations are observed and some mutations are not observed. I'm talking about protein amino acid change in amino acid, right? So this means the job is really difficult for any software. And AI, so now we have lot of data where we know that change is there but no phenotypic change. Change is there but mild phenotypic change, and change is there and huge phenotypic change. If millions of such amino acid changes, we have the data, train the model and let the model tell that if given this behavior, what is the likely outcome? And that's where they have got into it. So this means people have started using AI, although bioinformatics tools existed, but now AI tools are performing better than that. Okay, so that was a summary. How much time I'm left with? At what time we have to stop? 1:15. Okay, so I have 45 minutes roughly, right? Yeah, fine. So moving on. So I'll talk about one AI application, very recent AI application. Okay, and which is in this particular area, which is alpha genome. So while some of you might have heard of AlphaFold, okay, Alpha Missense, but this is a recent tool called Alpha Genome. Okay, so Google has a division called DeepMind. Okay, and they are the ones who are developing it, and we'll slightly look into it in details, right? So you can visit this website. Okay, I'll share these slides to you. So what is their web page? This screenshot is from there. It says that introducing a new unifying DNA sequence model. So it's a model that advances regulatory variant effect prediction and promises to shed new light to genome function, now available as a API. Okay, something which we can communicate to their server. Okay, so this means so such a tool is available, and they can find out the variant effect. What is the effect? And this can be used to predict whether there is a disease risk or not. In drug discovery pipeline, such mutations can be annotated, whether this, while target identification, which protein I should, for drug discovery I should target. Okay, so for target identification, which site should be targeted? To mine those things, which site should be targeted? He mentioned one thing, the active site, the residues from active site can be important, right? It is indeed important, but active site is not just made of one site. It is made of many amino acids, and right, so this means among many amino acids which can have a high impact, right, which can have more impact. So this means this tool can be used over there, and you can tell your students that, look, now there are the apps, okay, which can annotate among the in the protein sequence. It can tell that any variations in a protein sequences, how impactful it is, right? And so if you want to look into it, how it works, again I have copied this one because they have summarized it very nicely. So currently they work with a window of 1 million nucleotides. So you take a genomic length of size 1 MB, okay, 1 million nucleotide bases, okay, that much. If you take a DNA fragment of that long from genome, right, and then you give it to alpha genome, right? I'll also tell you how this works, right? So then what kind of output it will have? The kind of output it will have, it will generate a range of data, which is shown here. What is written here? For example, what is the top track? Can you tell me? Yes, what's your name? Dharm. Sitaram. What is the first track? In the output, what is the first track? So I told that they will give you a range of data. What is the first data that it has given you? Gene expression, this one, right? So here they have given listed some of them. So gene expression, and then there is a track also. It tells that. So this means how it is interpreted. It tells that. There is an amplitude, right? So high amplitude means more expression, and blank means if it is empty, this means that no expression. So right now this alpha genome is giving the data for this window, right? So for this window they are telling that, so this means that at this position in this region there is some expression, in this region there is expression, this region there is expression, and otherwise there is no expression. Not only expression data, okay, it also provides you accessibility data. Means DNA accessibility data. It provides you histone modification profile. Why histone modification? Histone modification has a signature. So those who, do we have any teacher who teaches epigenetics? Gene regulation, you teach gene regulation, right? So gene silencing. So yeah, so of course you have to teach that also. So the kind of modifications, histone modifications, okay, whether methylation or acetylation, at which residue it is happening, it has a particular meaning, right? So this means histone modification profiles are very important, which tells that whether the DNA will be, that region will be condensed or it will be accessible. Okay, so this means accessibility report, histone modification profile, TF binding. So from ENCODE is a database, around 10 years back it came, okay. So from ENCODE database, they also have information about transcription factor binding, where all there is any binding to that region. So for each nucleotide you see what is the profile you have, and then splicing information, okay, whether there is any alternate splicing or not, right? And then chromatin in contact map. So those who again teach epigenetics, I think they must have heard about a technique called attack, seek and other things, where so within the genome, which region of chromosome 1 is interacting with chromosome 22. So this means chromosomes are not very disjoint, okay, just like the way you are sitting, you all are not sitting as an individual student, not having any interaction. No, there are interactions that I can see, right? I saw the interactions over a cup of coffee, okay. So just now there was a tea break, and I saw that in smaller groups you all were interacting with your the group member, okay. It doesn't mean that you all were from same colleges, right, or same department, not necessarily, but there were interactions. It need not be they all can be from same chromosome, they can be from different chromosome, and some interaction is happening. So there is a common theme, some relevance, some importance is there. So this means they also obtain, okay, they don't generate it, they extract this information from publicly available data. So first they will create a genome patri, DNA. So it's like DNA genome patri. For each nucleotide they will get a profile. It is like these days you must have heard in the news that Trump has really gone crazy and it is behind Iran, Venezuela, okay. And so what are they doing? I mean, they are creating a profile, okay, who is their target. If let's say Iran's that supreme leader is a target, they are profiling him, all the intelligence report they are collecting, and then they are hitting it, right? So this means look at the kind of I mean, they are crossing boundaries and they are collecting that much intelligence report, and then somehow they are able to capture him, whether Venezuela president or whether the supreme leader of Iran, they are able to hurt him and they are able to capture him, right? What ensures the success? The success is ensured because they have a very detailed profile of that person. Similarly, for each nucleotide, they first generate a profile, all possible information. Only a subset of them are shown here. Many more information they will generate, right? And then they have already been trained. The model has already been trained. So now they can, based on this behavior and based on their previous training, they can tell you what is important, what is not important. I'll show some slides. So this alpha genome, if you go to GitHub, I have given you the link where this package, AI package is available. So this package is freely available for anyone to use. Okay, you can use it for, if your student is interested, they can. Oh, by the way, so far I have not used it. It came last year, but still my students, they have not used it. Me as a faculty, I am yet to apply it in my practical class. We have not yet done it, right? But then it is not very difficult to do it. All you have to do is that if you want to use the API, so you have to use, so get the API key and then you can communicate to their server, okay? Or you can download it. So you can download the package from here, okay? They give you all sort of instructions, okay? So for example, you can get the data, you can get the package from there. Now the problem is you need some hardware requirements that not all of you may be having, but your university or your related connected university may be having it. For example, you may need a very high performance NVIDIA H100 GPU server to perform such operations. Okay, so you may need a lot of data. Okay, because a lot of processing happens. Okay, these are AI tools. If you are trying to analyze your own data, then you may need all those things. Okay. And since it is written in JAX, so JAX installation and then TensorFlow. Okay, so these are, so some of the model requirements are there, hardware and software requirement. Hardware requirement can be challenging, software requirement not that challenging, right? And then some pre-trained models, they have already kept it at Kaggle. Kaggle, one of your previous teachers, they might have shown you that in data science, not only the biological data, lot of data we use in data science, all of them are, and most of them are publicly available at Kaggle. Okay, if not there, then at Hugging Face. Okay, so some of the pre-trained models have been kept there. Okay, but typically how it works, right? A simple Python program can help you to get the job done. So you can see that. So here is the okay. Okay, so you can see that just load certain packages. So load alpha genome data visualization research model. Pretty obvious. It is from their GitHub page. Okay, I just wanted to tell you that how easy and how difficult it is to do. Right. So this means all you need is that they have developed code. Okay, and all you have to do is that use it in a very simple manner. So here they tell that they select a model. So this statement, they create a model, they choose an interval, which region of the genome, I think they have done it for human genome, so which region of the genome they should look into it. So you have to give a window that in which chromosome, let's say 22, and from here to here you have to look into that, right? Once you tell that, and then you have to also tell that are you interested in a position, so variant. So here you have to create a variant profile, okay, by using a program, by using a method. Okay, so we tell that in chromosome this at this position the reference base was this and alternate base was this. So you have to first give all those things, and then the output will be stored in this particular variable, and these are the things that it will store. And once the output has been extracted, you can plot it using a sample command which is shown here, right? So this means these are pretty simple steps. Okay, and it will communicate to the server, fetch all the relevant data, and it will give you, it will tell you the relevant information. No, right now this is for human genome. They have developed it for human genome, but can be scaled. I think the problem with this is, I think when there is a heavy graphics, I think it creates a problem. Yeah, so Colab you can try that. So better use a API. I think give it a try. Okay. So on Colab they have kept their API, and through Colab, so Google has kept up. There is a platform called Colab where any programming, whether you are interested in Python or C or any programming language, you can use that platform. They have a compiler or interpreter, and you can use that platform to do the coding, right? For alpha genome, Okay, so they have given you a sample code. Okay, and through Collab you can use it. I think you will have to get a API key, and once you have the API key through Collab, you can get the API key also, right? And then because for Collab you'll have to log in into Google, right? And this is a Google package, so the through Collab you can extract the API key, and then you can do all the analysis. So I'm not able to show that, but what I wanted to show you, maybe I can draw that. So some when you will see the slides, you will find that there are so it is expression data. I think the sample that they have taken was colon. Okay, so it is a cancer colon transverse section. RNA-seq data means transcriptome sequencing data, means expression profiling data, right? So, so it is expression data, patient 1, patient 2, data set 1, data set 2, right? And then what it shows that the region that this tutorial had selected, right? So a genomic region. So this means along this length, what it will show is the gene structure. Okay, in the genome, in a region of a genome, a particular gene. So for example, you can say that this is UTR, first exon, second exon, third exon, and something like this, right? So this is a region of the genome, and then they tell that, so those individuals, okay, where there was, who had one particular allele, what was the expression profile in that region, right? So for example, let's say, so do I have colored markers? Yes. So let's say in the wild type, those who were healthy, so let's say the expression profile was something like this, right? But then those who were mutant, okay, where how was the expression profile? So let's say it finds that right. So now you find that between an individual having a allele, let's say, so such individuals had a allele, let's say A, and such an individual had a allele, let's say C. Okay, so when I say C means, let's say at this position, so at this position there was this individual, one individual had A, another individual had C. So in this region of the genome, right? So there were two alleles. Now there is no other change, but two individuals having two different alleles, and look at their colon expression profile. Forget about, let's assume that it's not a cancer sample, just a expression profile. I think when there is a heavy graphics, it creates a problem. Right? So how the alpha genome is trying to interpret based on the training and other things, what it is trying to interpret is that because of this allele, what is happening because of this allele, whether in the regulatory region, whether it is in intronic region or genic region, it is telling that the gene expression of that gene is getting diminished. This is the impact they are going to identify. So I deliberately kept a case where I can give you a feel that how from concept, from for a classroom purpose to the lab, okay, how the story unfolds, okay, how these kind of things are being used. So this means this is how alpha genome works. This is how it tells you the impact. So in few slides, I mean some of the preceding slides, we talked about the variant. We discussed about the variant impact, but now it tells you that it has predicted the impact, that because of that allele there is a change in the mutation. And in the case, yeah, just hold for a second. When the slide, maybe the issue with the slide, leave it. No, don't worry, I have explained it, don't worry. Because of the heavy graphics could be the region. Or can you load the PDF file? Can you load the PDF file? Maybe that would be better. There were 2 files, PDF file. AI in biotechnology, yeah. PDF, yeah. Go to screen. Slideshow mode. Full screen. Yeah, it works. I think there was some issue with the file. This is what I was trying to show, that they have done it for 3 samples, okay, and here they call it what is a reference allele and alternate allele, labeled by gray and red, okay, and then here they show that because if the allele is gray, then look at the expression profile. So this is the genomic region, okay, and this is the gene body which I was trying to explain here. Exon, UTR, this may be the 3' UTR, exon, and UTR, right? And then it is telling that, look at the expression profile, okay? 3 different transcriptome samples, and look at the difference between gray and red. Gray represents the healthy, I mean having the reference allele, and the red represents having a different allele. And because of this mutation, and so this allele is located over there, you find that wherever there is an exon, there is a difference in the expression, right? So that allele is having that much effect. It is having a regulatory effect, which it is able to predict very correctly. And not only that, it is also telling that, look at in these 3 samples, this exon is not at all expressed. Do you see that? So this exon, first exon, it is not at all expressed. Only gray curve is there. No red curve is over there. So it also has predicted that there is an exon skipping, means alternate splicing. So one mutation, it is having so impactful that it not only alters the expression level, it also alters the isoform, transcript isoform. So this kind of information it can process and serve you so that you can use it and you can proceed with whatever, I mean for the downstream things. You have to provide 2 inputs. 2 inputs means, so basically what it does, that for the genome region of interest, if you specify that, right, and if you specify genome region of interest and you specify the position of interest, if at all, it tells that there are, for example, there is a variant, there is a position where you find a SNP. You are interested, you suspect that that SNP could be very important, right? So you can tell that in this window, you tell me that whether this SNP, what is the impact it has. So that information you have to provide. Means what is your interest? Human genome has 3 billion bases, right? So you have to come out with that, what is the position of your interest or what is the region of your interest. Then it will take the flanking region and extract all possible data, generate a chunk of data for the region of your interest, and then for the base of your interest, it will do the prediction. So this is how it works, right? Now we are left with only 15 minutes, so we'll have to quickly move. So now let's wind up the first part and let's delve into the another part, which is protein structure modeling. Okay. So I think tomorrow there are going to be 2-3 labs on that, and maybe 1 theory class on protein structure modeling. Okay. So why modeling? So it doesn't. So why modeling means how do we obtain a protein structure? Anyone from this side would like to answer? How do we obtain protein structure? X-ray crystallography or NMR or other biophysical techniques are there, right? So this is how, so means first we have to crystallize it and we have to get the structure, right? But then this is not easy there are millions of bacterial prokaryotic proteins, eukaryotic proteins, plant specific, animal kingdom specific. Millions of proteins are there. Okay, it is not possible to crystallize all of them and go for the X-ray structure. Maybe there will be a time when that will be possible, but for now it is not possible. But often, for example, just now we talked about this gene APOL4. Okay, what if the protein, so now we know that there is an interesting position, right? Maybe what if it overlaps with the coding region? Okay, there is a SNP which is causing impact, but and what if we want to go ahead with drug discovery or finding an inhibitor for that, right? But unless you have a protein structure, how can you do that? That's where, so if we can't get the protein structure experimentally, then we can model it. And how to model it? By learning it. There are certain scientific principles, algorithms, bioinformatics principles through which it used to be done. There are a lot of tools. Okay, there is one very classical technique called homology modeling. So if the structures are known for similar sequences, okay, then you adapt that. Okay, it's like let's say someone is going to start a university. Okay, the government decides to establish a university in Nalgonda district, right? Now already in Telangana region there are universities. So what if a similar set of courses are going to start? We can learn from existing university what should be the structure, and maybe to some extent we can copy it, but no need to create a duplicate, but at least we can learn major organizational structure, academic structure, all those things we can learn. The same thing it is being done during the modeling, right? It is being done. So what is the problem? Is there a gap here? Was there a problem? Yes, there was a gap. So there was so the structural biologist, they had a community called CASP, which is a critical assessment of protein structures. Okay, so CASP group, it was nothing but it was a competition. Okay, like hackathon and those kind of things. It used to be a competition. Okay, so periodically they used to, what they will do, they will solve structure of some new proteins, right? And then they will solve the structure, but they will not make it public. And they will tell that whatever the technique you have, you come, they will give the sequence and tell that you give the structure. And during the competition, they will come, they will, the sequence will be given, they will optimize, whatever you want to do, you do it. And then you submit the answer, you submit the structure. And then they have the very high quality at the very fine level, angstrom level. Okay, we have that resolution, we have the X-ray structure. Then they will overlap the two structures, and then they will say that what is the accuracy, 50% accurate, 60%, 70%, 80%, 90%, right? Their target was 90% and above. If they reach it, they will say that, oh great, they have done a success. So that's why many teams, many researchers, they used to participate. If they used to think that, oh, my algorithm is better, they will participate and come out and they will say that, give me the sequence, I will solve it. And they used to solve it. I mean, they will predict it. They used to predict it, and then the CASP team, they will evaluate it. But the success did not cross 70%, 60 to 70%. That's where it hovered. 60%, 65%, 63%, this optimization, that optimization. But there was a twist, and the twist was brought by this tool. Okay, I mean, if you want to quickly go into the history, so in 2018, this team, the DeepMind AlphaFold team, they participated in this competition, CASP 13. And then later in 2020, they participated in CASP 14. 2020, you remember, November 2020, COVID time, right? During that, they were not working in lab, they were working from home, and still they were competing for that. So 2018 and 2020, in both the cases, their predictions were, it outperformed all existing algorithms, whether Rosetta or you take any other algorithm. Rosetta is one of the good competitor, right? So this means they were very excited by their performance, okay, and they also published their algorithm. And their idea was what? So far, the researchers, the protein chemists, they used to think based on the protein chemistry, the interactions, all the chemical interactions and three-dimensional interactions. So from physics perspective and the chemistry perspective, okay. These people, they thought entirely different from the data perspective. They looked at millions of the structures, right? And they learned that if this is the sequence, in what is the angle, what is a torsion angle, this angle, that, I mean, alpha and beta, all those angles you may remember from your textbook knowledge, that what are the various angles that it forms, okay. Sorry, alpha and beta sheet will have different torsion angles. So they used to so they used to estimate that. So their approach was data oriented. From all the existing protein structures, if this is the sequence, what kind of motif, what kind of arrangement it will make. So they converted the known structures into a trainable structure. So you must have learned in the first 2 days how the data is, that not only data cleaning, how an image is converted into a data which is compatible for training, right? So this means that data transformations and data conversion is also important. So this means their three-dimensional structure was converted into appropriate formats, which was used for training the model. And they trained it, and then they had huge success, okay. And given that success, so in 2021 they published their methodology, okay. And then they launched a database as well as, so they launched a database as well. Means whatever the structures they had predicted, it was already deposited in the database. And you know what was their accuracy? They reached 90%. So this means they could reach accuracy of 90 and more than 90% for the proteins which was being tested. That was a performance. That's why suddenly they were in news, okay. And then finally what happened, they launched AlphaFold 3, and then last year this team was awarded. So I mean, 2 people from that team, they were awarded Nobel Prize in the area of field of chemistry as well, because of their immense contribution. They solved one big problem, okay. Just by sequence, their structure were as good as the X-ray crystallography structure. That was the power, okay. And now it is a global research tool, and this tool is available. So I don't know whether that will work or not, but a server is there. In case if you are interested, you can submit your sequence and you can generate the protein structure, okay. And then I'll come to that. And if at all, and for existing sequences, those which are already there deposited in protein databases, you can search I mean, there is a search box. You paste it by IDs or anything, and based on that, you can fetch the already predicted structures, right? So they have a database, they have a server where you can generate your own, okay. If time permits, I think we have few minutes. I would like to show you, but whether this works or not, I'm not sure. I think that is lost somewhere. But, are you able to see that? Okay, so it won't load in that.
We've discovered more about the world than any other civilization before us. But we have been stuck on this one problem. How do proteins fold up? How do proteins go from a string of amino acids to a compact shape that acts as a machine and drives life?
When you find out about proteins, it is very exciting.
You can think of them as little biological nanomachines.
They are essentially the fundamental building blocks that power everything living on this planet.
If we can reliably predict protein structures using AI, that
It has come. Okay.
can change the way we understand the natural world.
Should I start from?
Protein folding is one of these holy grail type problems in biology. We've always hypothesized that AI should be helpful to make these kinds of big scientific breakthroughs more quickly.
And then I'll probably be looking at little tunings that might make a difference. It should be creating a histogram one and a background histogram.
We've been working on our system AlphaFold really hard now for over 2 years. Rather than having to do painstaking experiments, in the future biologists might be able to instead rely on AI methods to directly predict structures quickly and efficiently.
Generally speaking, biologists tend to be quite skeptical of computational work, and I think that skepticism is healthy and I respect it, but I feel very excited about what AlphaFold can achieve.
CASP is when we say, look, DeepMind is doing protein folding, this is how good we are, and maybe it's better than everybody else, maybe it isn't.
We decided to enter CASP competition because it represented the Olympics of protein folding.
CASP, we started to try
I was talking
and
about this
speed up
CASP.
the solution to the protein folding problem. When we started CASP in 1994, I certainly was naive about how hard this was going to be.
It was very cumbersome to do that because it took a long time.
Let's see what we're doing still to improve.
Typically 100 different groups from around the world participate in CASP. And we take a set of 100 proteins and we ask the groups to send us what they think the structures look like.
We can reach 57.9 GDT on CASP-12 ground truth. CASP has a metric on which you will be scored, which is this GDT metric.
On a scale of 0 to 100, you would expect a GDT over 90 to be a solution to the problem.
If we do achieve this, this has incredible medical relevance.
The implications are immense, from how diseases progress, how you can discover new drugs. It's endless.
I wanted to make a really simple system, and the results have been surprisingly good.
The team got some results with a new technique. Not only is it more accurate, but it's much faster than the old system.
I think we'll substantially exceed what we're doing right now.
But then it's a game changer, I think.
In CASP 13, something very significant had happened. For the first time, we saw the effective application of artificial intelligence.
We've advanced the state of the art in the field, so that's fantastic, but we've still got a long way to go before we've solved it.
The shapes were now approximately correct for many of the proteins, but the details, exactly where each atom sits, which is really what one would call a solution, were not yet there.
It doesn't help if you have the tallest ladder when you're going to the moon.
We hit a little bit of a brick wall since we won CASP. Then it was back to the drawing board and like, what are our new ideas? And then it's taken a little while, I would say, for them to get back to where they were, but with the new ideas. And then now I think we're seeing the benefits of the new ideas. They can go further, right? So that's a really important moment. I've seen that moment so many times now. But I know what that means now, and I know this is the time now to press. We need to double down and go as fast as possible from here. I think we've got no time to lose.
So the intention is to enter CASP again. CASP is deeply stressful.
There's something weird going on with the learning, because it is learning something that's correlated with GDT,
Anyway, you
but
can
it's not
watch
calibrated.
this story.
I feel slightly
It will take long.
We should be
Finish it
learning this
in a minute.
in the blink of an eye. The technology advancing outside DeepMind is all.
Because it will take another 5 minutes. Okay, and now in last 1 minute, we'll talk about, so can AI predict the impact of mutation on protein structure? And To some extent, we also talked about this when we were talking about alpha genome, right? So I give some insights, and I think there is a specialized tool called alpha missense, okay, which is a specialized to do primarily this particular thing, whether there is a mutation in the protein structure, right? Whether it will have an impact on the structure or not. So here, this is a structural perspective, right? That the earlier it was a genomic perspective. It was a structural perspective. This has a structural perspective, and yeah. So just I wanted to introduce that, and this particular the last slide, the AI application in drug discovery. Okay, so this I copied from one of the recent reviews by Zhang et al. From Nature Medicine, which tells you that drug discovery is not just a one-step thing. It's like you discover, but rather it is a pipeline. There are many steps. Okay, maybe Dr. N. Prakash Prabhu will also throw some light on that, how AI is being used. Okay, but broadly we start with a target, often a target protein, and then drug discovery, some preclinical studies, which also involves safety aspects. So when we talk about target identification, so what is the protein that we should be targeting? And then drug discovery means what is the lead compound? Okay, so screen the molecules and what are the lead compounds. Then the safety aspects, okay, comes into picture, or ADME, absorption, digestion, and assimilation. All those ADME aspects come into the typical pharmacokinetic things come into picture. Then comes approval, and then the post-market surveillance. Okay, so this is a big, it's a long journey, but then see where AI has been intervening. I think based on our discussion that we just had, I think one thing that you must be clear that for target identification, definitely AI has contributed a lot. So if it is able to tell that there is a SNP which is having a very high impact, those can be some of the target proteins, right? So this means that there are many approaches, not just that, okay, but many approaches are being used to identify a target. And another challenge is that screen the chemical libraries. So we have a lot of libraries, and we have software tools also. They routinely used to screen it, right? But still there were certain gaps, and this data science based approach, it is outperforming the existing approaches, existing bioinformatics algorithms, right? So which is again being heavily used. So in this area, in these two areas, it is used a lot. And when it comes to safety aspects and preclinical trials, again computational models are there, and again AI is competing over there with their models. Okay, so this means AI is given the data, and particularly the drug companies, the pharma companies, for the previous drug discoveries, they have a lot of data. So very curated data, good quality data. Using good quality data, they can make better models. And the drug companies, they are collaborating with many people. So like pharma companies like Eli Lilly, or you have Novartis, or you have many pharma companies who are in the drug discovery. They are teaming with ChatGPT, the one who has developed these large language models, okay, or such language models. So they all are collaborating with each other to find the solutions for these particular steps, right? So I think my job was to just introduce you about its application in genomics, protein biology, and drug discovery, and there may be specialized sessions on that, which will actually the lab sessions will give you more better idea than just the theoretical ones. So with that note, I would like to stop here. Any quick question? So any quick question? We can have one or two questions, otherwise yeah, we can also discuss over lunch. Fine. Yeah. Is there a laser pointer also? अन्ना लेजर पॉइंटर है हाँ okay, so can we start the session? Difficult session, post-event sessions are usually difficult for not only for resource persons, I think for everyone. Okay, so I think so while we talked about many aspects, but one of the lab that was listed in the program was identify microbial species from sequencing data sets, right? It's a very classical problem and people have been working on this. Do we have any microbiologist here? Microbiology teacher, you are microbiology teacher. Okay, so typically if this has to be done, okay, how a particular microbial taxa is identified? What, classically, I'm not a microbiologist, yeah. You have identified based on the colony characteristics, okay. You go for, so this means that morphological characteristics and culture based characteristics are not too accurate, right? So what do you do at the genome level? You do ribosomal RNA sequencing, right? But let's say you have, yeah, so you have let's say cloned or amplified ribosomal RNA sequences. Then how do you identify that? I think you are pretty, I think you have already told the answer, or you also added to that, okay? So this means, so I was asking, what are the classical practices as far as microbiologists are concerned, how they identify it, right? And they said that, yeah, if they can culture it, then they would like to characterize for various biochemical and morphological aspects, but not all the time they are able to pinpoint what is a taxa or what is a species or what is a genus that sample is. And she suggested that we can go for some sequencing based approach to do that. And I think he already gave a clue that it is a 16S ribosomal RNA, which is one of the marker gene. Why marker gene? Because it carries enough variation so that you can distinguish, if not two species, two genus, or at the general level you can classify. But at species level, 16S sequences, not all the time they work. In particular when it is itself incomplete, because when we do PCR, how long we can amplify? So when we go for PCR amplification and sequencing, so typically 300, 400 is a typical length that we amplify and get it sequenced. 300, 400 nucleotides doesn't have enough information to distinguish at the species level or strain level, right? Now, coming to the okay, so this was as per the program. It was given that data sets, so one can work with metagenomic data sets, tool, NG Rust and activity. These are the activities. So this activity I have added. Now, as far as metagenomic data sets, so I think so far our discussion was very clear. There is a microbial taxa we have to identify. Now, what is a twist that I don't understand about this kind of data set? So, any microbiologist or non-microbiologist, they would like to throw some light. Now, what is this thing? So far, we were talking about how to identify a microbial strain or a microbial species. But then what is this? We are, anyway, we all are learning. Yeah. Any input, any. Sorry? You did not. get. I'm saying what is this data set then? Yeah. Why we are deviating from the line means instead of microbial identification, why are we now talking about this data set? Okay, so it has a strong connection in microbiology. You want to add anything? No? Yeah, so he is suggesting that we can identify microbial community. Okay, so this means that now there is a new word. So far we were talking about a microbial species. Now we are talking about a concept of ecology, which is community. So there is a microbial community, and where it is, so in a microbial community means there are many members, right? So compared to something that is being cultured, so you have a strain, you are culturing them, you are characterizing them. You have a community, you are not able to culture all of them, and then you have to identify. So this means in a culture, culture-based approach is much better compared to this approach, which is your community-based. So this means in a community, if there are many people, right? So for example, during the morning session, I was asking who all teach genetics, so or genomics. So at least I remember one person. You said that you also teach epigenetics and regulation and other aspects. So I remember those two people. What if there would have been a formal introduction round and you all have shared your expertise? Was it possible for me to remember in just 5 minutes introduction about all of your domain area, teaching area? Not possible for me. Similarly, 1 or 2 microbial species, we can characterize it in depth, we can do that. But when we have a community, means there is a challenge. Clear? So this means it is more important to identify, it is more challenging to identify a species in microbiome. Microbiome means microbial community than one individual which is being cultured. So anyway, I'm trying to set the background. Okay. By the way, how much time do I have? Can anyone tell me? 40 minutes I have. Okay, fair enough. Not bad. So from microbes we reach to microbial community, and from microbial community now what is metagenomics? That is not clear. Microbial clear, microbial clear, this is not clear. Now there is a connection. I think he tried to link that. Okay, thank you for that. The connection is, now we have a microbiome, we have a microbial community. Many members are there. Their relative population size differs. In this group, there may be there are faculty members from different colleges. Few colleges may have more than one member, right? Few colleges may have nil member. AP has so many degree colleges and professional institutions, right? So this means the relative population varies, right? So similarly, in a microbial community, it varies. Now, what is the concept of metagenomics? So meta means beyond, and genomics. So it's like the classical genomics have been identify an individual, identify a species, and work on that. This is like going beyond that. So when we go beyond that, means when we have a community, the concepts of genomics becomes even more complex, and that is a concept of metagenomics, where you are not talking about genome of one individual. Okay, we are not talking about genome of one species. We are talking about a genome of many individuals, many different species. They can be proteobacteria, they may be formicutes, maybe unicellular eukaryotes, right? It's a community we don't know, right? All microbial species, prokaryote, eukaryote, viral, everything would be there. And then we are talking about characterizing them, right? So there is, so same way what you and your fellow members mentioned, that we can use genomics tools, same genomics tool can still be applied. Whatever we do for one strain, okay, we can scale it up for entire communities. This means again at the community level, we can do the sequencing of 16S ribosomal RNA, or if it is eukaryotic, maybe 18S ribosomal RNA or intertranscribed spacer. There are many regions which is used, okay, not just one. We can amplify it, send to the company, get it sequenced, analyze the data, and then identify it, right? So this is how it is done, okay? Now, in this area, while this entire program is on application of AI in biology, but still, whatever the present tools are there, they don't still apply a lot of AI, right? And so moving on, let me okay. So as far as identification of microbial species, whether in a community or as an individual is concerned, we have two different approaches. Just now we talked about marker gene based approach, and these are the tools, existing tools, which can help us. Since being a lab session, we can focus more on this. But then microbial species can also be identified by alternate ways. For example, and that is, instead of sequencing one target gene, right, one can sequence entire genome, okay? So it's like if I have to know more details about him, instead of just asking what is your name, okay, I simply ask the organizers or ask the coordinators, whosoever they are coordinating, get his profile, and then his profile tells that what is his date of birth, what are his parents' name, where he did schooling, where he did UG, PG, what is the first job he joined, and then what is the so many jobs he joined, where all, what is his ancestral place, native place, where he currently resides, the entire profile, everything about him. Still, I think I can identify that way. I can identify much better than the previous way. So it is like identifying something based on just one marker or by whole genome. The complete genome sequence can also be used to identify, but complete genome sequence can be used for many other purposes, right? So this means there are two, depending upon, there can be two data sets, and depending upon which data set we have, we can identify. Clear, right? So this is about identification of microbial species, right? In particular in a community. Clear? Whatever is written, there are many tools. I have just listed 2-3 tools. Can be online, can be standalone. Standalone means you need to download it, install it, and run it, right? So it may differ. For this also, whole genome sequencing data, anyway, both are sequencing data, but more complicated, and there are different set of tools which take care of this, okay? Metaphylan, Kraken, and mgrast, but mgrast is common in both. So mgrast is common in both. So when I was informed about this lab session, I thought of creating some accounts, but unfortunately even I had an account at this portal, okay, even I was not able to access my account for some reason. Then I thought of I'll create some dummy account, participant 1, 2, 3, 4, 5. Still it gave a problem. So looks like it is having some issues, okay. So and why we need an account over here? We need an account because once we have an account, we can access any data set which is made public over there, use the data, and we can analyze it and learn it, right? Whatever the purpose is to identify the species. So this means for today's session, this is not going to work. So this means we'll have to find an alternate ways, right? So there are alternate ways as well. So we have online server called Silver, okay? And as you see the summary, a comprehensive online resource for quality checked, curated, and aligned ribosomal RNA sequence data. Mostly 16S, 18S, and then maybe, yeah, and then the larger subunit data as well from the 3 domains of life. So mostly it contains ribosomal RNA sequences from the major domains of life, okay, which is listed here. Fine. So this means this is a database, and not only it is a database, it is a server also, and today we are going to learn, we'll practice this only. Fair enough, right? So Yeah. As far as phenotype prediction is concerned, if we are able to successfully do it, we'll get to that. For the phenotype prediction, that is an area where a lot of AI is currently being used. Okay, and in my lab also, I'm working on one AI solution for that. So, so as far as phenotype is concerned, I think microbiologists, they know that what are the morphological features, what are the biochemical features, is it an anaerobe, is it an aerobe, is it a obligate parasite, it is having any symbiotic interaction. Means there are many things that we would like to know about it other than the taxa, right? There are many interesting information one may be interested in. Okay, whether it is as far as if it is a pathogen, is it resistant to any microbial antimicrobials? If it is a fungal pathogen, is it resistant or non-resistant? If it is resistant against which bacteria? Can we identify as much as information we can from a minimal information? That's what the predictive biology is, or AI is, or any other tool is, right? With minimum information, what is the minimum information which can give maximum details, right? So we'll get to that, but today we'll practice Silva. Okay. So if we have to practice it, so this means we are going to use Silva to do our job. Okay. Now let me go to their website. The link is also given there on the bottom. Can you see that? Okay, so what I'll do, I will go to, let's say if this is working, looks like it is working. So we can go to the Silva portal, web portal, just by searching Silva. I don't know, for some reason it's very slow. I'm not interested in this then. Okay, so the culprit is this keyboard. This is not responding. Okay, so this keyboard is not responding. So this means I'll have to face you like this. That may work. Okay. This is very annoying. Your computers are working fine? Yeah, this is frustrating. Okay, here we go. Are you able to see that? While we are waiting for this portal to load, we can also go to NCBI. Yeah, so we have loaded it. Okay, this is how it looks. Okay, so have you loaded NCBI page and Silva database page? Sorry, Silva you have loaded. Same way you can try NCBI, and yeah, this is how it has loaded, right? You can simply Google NCBI, or the URL you can see on the screen, ncbi.nlm.nih.gov. Okay. So what all you see on the screen? So anyway, this I have already showed you. This tells that what is a summary, how much data is already available. It further tells you that what are the key utilities available at Silva, or that Silva has to offer. And can you make out anything? Second last row, there are two microbiologists. Yes. What is this? There are three icons given there. Can you tell us what is the relevance? One says alignment, classification and tree service. Second says classification of NGS data. Third says Silva tree viewer. Can you tell about any of these? Any idea what is this? Anyone? Sorry?
Alignment is multiple alignment of
alignment of what?
sequence alignment.
Sequence alignment of what? Sequence alignment, there are tools for that. We are not here to learn bioinformatics tool called sequence alignment tool. Our purpose is slightly different. In that context, what is this? Right. So we are here to learn what? We are here to learn that from a metagenomic data, okay, can we identify the taxa? Can we identify the bacterial species? Not bacterial, maybe microbial species, right? That is our purpose, right? And this is a database of what? Sir, what? This is a database of what? What kind of information is present on this database? What kind of information is present in this database? Ribosomal RNA sequences, whether small subunit or large subunit, from all kingdoms of life. right? So this is a database which primarily contains ribosomal RNA sequences, right? So our objective is clear, and then what it contains, that should also be clear. So this means whatever we do, we are playing with ribosomal RNA sequences, right? Small subunit or large subunit, right? And so let's try to connect the dots. So typically, what kind of data set we have? So when we say metagenomic data, metagenomic data is no different from any genomic data, right? In appearance, you can't make out whether it is genomic data or metagenomic data. At the end of the day, these data sets are called sequencing data sets. To be more precise, these data sets are called next generation sequencing data sets. Why? Because classical sequencing is no longer used these days, except for some specific purposes. Okay, so this means so the data set will be in the form of next generation sequencing data. In short, they call it NGS data, right? fire this job, just search, for example, I'm searching lake market, right? I have done for lake market, Hyderabad, and searching for lake market. Why don't you search for something else that you are interested in? Which microwave community? Soil microwave community, rhizosphere microwave community, right? Skin microwave community. It's like or ruminaceous animal microwave community, right? Whatever microwave community you are interested in, you can search. You can search, but add I would SCENCES that. Okay. So the slide means is also very slow. Okay, I'm not going to use it, but can you search it? Or can you search it and let me know? So search for any microwave community, but maybe anti-consequency, or simply search for that, then select the anti-consequency. Did you get any result? Click search. Cross period. This is the single species, right? We are talking about microbiome, right? Because that's what we actually do. Okay. Search for any microbiome, lake microbiome, skin microbiome, head microbiome, hospital microbiome, but single micro, you can do that. I mean, but yeah, you can do that. But this will have to select for only the 16 days, right? Then if you are searching, then SRA is not that made for. Or whole genome sequencing. If you are sequencing 16S ribosomal RNA of one organism, then why should one go for next generation sequencing? That can be simply sequenced by Sanger, right? So that portal is not meant for that. That portal contains data set in GBs, okay, where millions of reads are there, millions of sequences are there for your sample, and usually we do it when we sequence one strain but fully. But if you are asking one strain and that to 16S, then maybe this is not the portal. Okay, let me know, have you searched anything? I'm coming to your seat. Okay, photosynthetic bacteria, that's fine. Now so on the left side, you can see that if you have selected the database SRA, on the left side there are some filters. Just like in Amazon we have filters. Show me the products in this price range. Show me the product from this company. Show me the product with this rating. Similarly, we have filters here on the left and right. Okay, whether are we interested in DNA or RNA or exome or genomic and other and something things like that, right? So things like that. Okay, so I think things are fine. Okay, things are fine. Let me try again so that you all can see, because I can find that all of you are stuck at different Okay, can you type after M? Can you type E? Yeah, and space and 16S. Right? Yeah, 16S. There is some problem, right? After that space. Yeah, then space rRNA. RRNA. Enter. So I was struggling with that only.
ఇంకోటి తీసుకోండి రెస్పాండ్ అవ్వట్లేదు ఏం లేదు బటన్ సరిగ్గా పనిచేయట్లేదు.
This keyboard is not anywhere you keep it doesn't work.
కొత్త కీబోర్డ్ ఇది ఏ బటన్ పని చాలా డిలే
Type anything A, B, C, D, F, G, H.
వైట్ కీబోర్డ్ ఇవ్వాలా
Get it.
Now, if you need any price system,
Okay. Yeah. So let me search. Okay. So we'll go to this. Where you have got some results.
For hospital microbial community.
Okay. You have got some. Okay. So for okay, so now you see someone has done 16S ribosomal RNA sequencing data, negative control, this control. You see now if we click on this, basically I want some 10 sequences so that we can test it. Okay, so if we click it, so it tells me about that data, right? I want to access the sequences, right? So to access the sequences, we'll have to go to this link. Right? No, it is not.
వీడియో పరిష్కారం నుంచి కదా ఇక్కడనే ఉండండి పెట్టేసి
We need to turn this on. Working then? Yes. Thanks a lot. Now we have 0 minutes? 5 minutes. Okay. Okay, we'll quickly finish. Don't worry. Okay, so you said that yes, you were able to access data sets like this. Okay, now if this is working, let's click this icon reads, right? And then it is showing us some data, right? So it is showing us data. So this means how many there are around 5,000 reads are there. 1, 2, 3, 4, 5. It is showing just one set of data. No, in fact, 2 sequences. What does that mean? This is another sequence. Okay, fine. So there are 2 sequences, and what does it tell? Usually when sequencing is done, at times it is done from both ends, 5' end forward strand and 5' end of reverse strand. So it is done in two different directions, so that's why we have two sequences, right? What we can do is, we can, let's copy one, okay? Although this format is not good format, but we go back to Silva data, right? And let's try to paste it, right? It did not paste properly. What we can do is, no, looks like this is fine, okay? Yeah, so we pasted it properly, okay? So this means we have millions of sequences or thousands of sequences. One sequence we have pasted it. We don't know about the taxonomy, right? And we want so this means we have pasted it, basic alignment program. So this means we have to align against the SSU. It's one of the smaller subunit, right? So because we already checked that it's a 16S ribosomal RNA sequencing, right? Bases remaining aligned at the bases should be removed. Yeah, so that should be removed, that's fine. Now what do we want? We want classification, okay? So we will select search and classify, right? It is asking for some parameters, that's all right. We won't bother much about it, we'll go with the default. And we'll say that everything remain as such, and everything remains as such, then somewhere we should have a run tool, right? And we should submit the job. So this means we have one query sequences, a microbiome will have thousands or millions of such sequences, and let's see for what is the answer for one, okay, whether it is able to classify it or not based on the parameters that it has chosen, right? And I have already shared the list name of the softwares. If all millions of sequences need to be classified at the same time, then you have to use that one. For example, CHIME, you can use CHIME, or you can use what you call if whole genome sequencing data is there, Kraken or Metaphilan, these are the tools. But and just for the learning purpose, we have submitted one job, and let's see what is the answer. Anyone who has received the answer? Anyone who, are you able to try that? Can you do that? If you have any problem, tell me. I mean, where are you stuck at? I could resolve my keyboard problem. Are you stuck at any place? What is?
We stuck at this place.
So you are stuck at this place. What did you search?
E. coli bacteria.
E. coli bacteria, preferably go to SRA. SRA has no hits. So this means SRA has no hits. Simply search for 16S microbiome or 16S metagenomic, something like that you search, and go to the SRA portal. Yes? Yeah? Now, so now is it amplicon sequencing or is it whole genome? Why don't you query box? No, it was fine. I think first you have to know thanks, or yeah, then select SRA. Any luck? Yeah, go to SRA and simply, what was hospital? You type hospital microbiome 16S. Simply type all this 16S, the name of the marker gene. Hospital microbiome and yeah, hospital metagenome 16S. Any luck? Now you can go to the, so is it a 16S sequencing data?
Yes.
Yeah, you can go to reads and select 10 reads or 15 reads or one read, whatever that permits you. You can go to Go down. You can go to this data, right? And from there, go to the tab called reads and download few reads. You can download these two, you can download and you can fire it. Yeah, these two, you simply copy and if you want, then wait. Because if we submit too many jobs, it will just affect our. Where are you? Amplicon, this is fine. Then, where? Let's select any, right? Right? Let's select this one. So now you can see how many, there are maybe thousands. There are 3,000 sequences out there. Copy one or two, or simply copy like this, right click, and then go to Silva and Control V, okay, and then choose what do we want to do? We want to classify it, okay, we want to identify it, right, the species. So we have to check search and classify. Yeah, tick that box, yeah, and then submit the job. Run tool, run tool, yeah, like this. Where are you stuck? If that is over, can anyone check what is that answer? Can anyone go and click there? Can you just check once what is that answer? Let's see what is the answer. Yeah. Now it is running. Okay. Are you stuck at any place? What is this? This is a home. No. So you can you display scores or yes. So this is the identifier. Can we scroll right to know what is the answer? Let's scroll right. Yeah. Can you click display? Yeah. No. Can we click on display scores? It should display as the final answer. How to get that final answer? So we have tested for one. Let me display classification. So because of can you type control minus minus? Where is control? Yeah, control minus minus. Okay, still it is not displaying us. Okay. Yes. So what we can do? Let's download it. Okay, can we open this? Okay, cancel. So now, can we go right? Can we scroll right just to know? Yeah, can we go right? So keep going right. Yeah, keep going right. Click somewhere. Yeah, and yes, keep going. Yeah, keep going. Yeah, wait. Okay, so this is, so what it has done, it has searched based on the parameters and it has given the full classification, and it looks like it is a gamma proteobacteria. So proteobacteria is a big group, okay, which has alpha, beta. So it is a gamma proteobacteria, and it is pseudo monadales, and then I think this is the Psychrobacter, and then I think maybe this is genus, and the species classification is not yet known, right? So it has classified till that stage. Okay, this is how it works. Where it is not shown? No, there because of the display on the page, right? Yeah, thank you so much. Yeah. Sorry, you said you have a difficulty? Yes, what is the difficulty? Yeah, so you click any of the data set. Let's first get some data. So this is our metagenomic data, right? We are going to that data. Ideally, you will have to download the entire data, and you will have to go to the. Do you remember we said that there is a Silva NGS thing, right? That's where you have to go. Okay. Now this is where the data will be located, and this is. Let it load. Okay. Let it load. Then from here you have to. I have shown that. From here you copy sequences and try it. Okay. Yes, sir.
Stop pops. This is something like from gut microbiome.
But there is no classification, right? So see.
No name is given. Sir.
So what did you try? From where you got that?
This one, sequence.
16S, okay. Of Alzheimer disease patient gut microbiome, right? You copied it and you pasted it here, right?
Sure.
And then now identity is 97%. So this means you should get some answer. Display classification, none. So it could not classify that. It failed to classify that, which is strange, huh? Yeah, you go and collect some reads. Yeah, and then whatever the reads, I think manually we can do 1 or 2 or 10, right? But there are automated ways by which you copy it, right, and paste it in that portal. Again, you are not copying it properly. Yes. No, give it one full sequence which starts from greater than symbol until here, right? This is one full sequence. We'll copy it and then we'll go there and we'll paste it. Right? And then we'll search the option of classification. Okay, so if there is 95% identity, then we will classify, right? And submit it. And then wait for the answer. I'm coming, 1 second, yeah. So see, so this is bacteria and unclassified. Then it says actinobacteria. So here is the classification. It has given you 2 classifications, right? Yeah. Yeah, coming. Following, what did you do? It says, okay. No, you did not select the full sequence, means here, the identifier also. The greater than this line. From the first line, yeah, that is the only problem. Which is below 95%, yes. Did it classify anything? It did not classify because you chose a cutoff of, when you were submitting the job, 95% was a cutoff. Go up, yeah. No, you did not, you did not give me this thing. You did not choose this option.
Okay, we have to search and classify.
Yeah, if you want to classify, then only. Otherwise, simply it will say that there is a match or not. But if you don't choose classify, then it won't tell you the taxonomy. Yes. Right? So, we can click this. It looks like this is the ribosomal RNA here. We can read it. So it is metagenomic, PCR, layout symbol, amplicon. This is the data. Right? We can go here. And then, since we are doing manually, go and select read. So these all are given here, right? You can select any. Right? You copy it. In fact, they have advanced options. No, that's all. No need. Copy it and submit to the Silva portal. You can submit many as well. Did you get it? Yeah. This is a tumor protein P53. It's not a
16S.
no. I think we'll have to search more properly. Not in the gene.
Page this, sir.
Silva database. Go to the Silva under classification. Silva portal. And then type whatever hospital microbiome or whichever microbiome.
Clarification for MDs, right?
Yes. No. That requires account. Right now, we are doing it manually. Go to the previous one. No. Don't close anything. Go to the yes. Yeah. And click this one. Because we are doing one case at a time. It will load. Yeah. Now paste the sequence and choose option of search and classify. Paste your sequence over there on the top in the query. Go up. Yeah. Input data. Yeah. Go down and then choose option of search and classify. Keep going. Yeah. Search and classify. There is a checkbox there. Yeah. Select that. Below that. Select that. Yeah. And then submit the run tool. On the right side. On the right side there is a right. And then let it run. Is that fine? So now I think it's already 3:30 or 3:25 only. 3:43. They don't need tea break. You can call the next speaker. Sorry. I think time permitted this much only. Any question? Any quick question for me? I'll share these slides. Okay. Your best teacher is anyway. You know who is your best teacher? Forget about practice. Those are things which is. Who is your best teacher? Yeah, your chatbots. Use chatbots. They can be better teacher, better resource person than me. Okay, ask them. They can guide you with these things. But I have already given the list of tools and softwares. Okay, I have demonstrated also that how it works on one sequence. For metagenomics, if you go to this one, MGRAST, it can do for all, and not only MGRAST, any tool can do for all, right? This is how it is being done. Yeah. 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 Oh, but don't trust ChatGPT so much for those things, right? You need to verify that. Yeah. That's fine. It might have mined it, but you need to verify. Ask ChatGPT, what is the evidence? How did you decide that? Give all the details. Then it will provide you the details. And then go through the details. Does it make sense? So he used ChatGPT to do the taxonomic classification, which is fine, and ChatGPT gave answer. But then we need to ask ChatGPT or any chatbot, give the details on what basis you have inferred the classification. Fine.
Perplexity is better. Perplexity.
I don't know.
Perplexity is very similar to ChatGPT. It gives you reference.
If there are no more questions, can we break out for tea? Because I think there is one more session. Who is the next speaker? Have to start the process. Yeah.
Equally.
right? So if you want me to repeat about the NCBI, you had a class on NCBI and how it works. So should I repeat or we can skip this? You are already done, right? Sorry? How to? Yeah. Basically NCBI, okay, I will just spend a very little time on NCBI. See, basically NCBI is a database. Okay. So this has 4 categories of data. Mainly it was actually started for the literature, which you call it as a PubMed Central. So the open source data will be available there. It has health data, that means most of the clinical data would be there. And then there are genomes, genes and proteins. Probably you would have had a class on this part only, right? Suppose if you have a gene sequence, you can compare it with other gene sequences. Or if you have a protein sequence, you can compare it with other protein sequences. Was that done already or no? Okay, maybe we will quickly do that. And the, NCBI also holds a small molecular database, though the basic idea was started with larger molecules or biological molecules. But of course, when it comes to the drug molecules, we need to have the small molecules. So that they host it as a PubChem. So these are the 4 major areas. Of course, if you go into that, there are a lot of further categories, but these are the major 4 categories where NCBI is hosted. So mainly, as I said, part of this is biological sequence analysis. So you can just type in Google NCBI BLAST. So it will take you here. If you type NCBI BLAST, alright. Do you have any common mail ID so that I can send some sequence which you can quickly demonstrate? No common. Oh, okay. How would you? Can you add me or I can take one of your number, then I can share with you. You are in that group? No, you are not in that group. Yeah. Uh, 93 938 44 6 11. Okay. Yeah, okay. So I will share the sequence, so you can share with everyone. Okay. Oh my God. You cannot connect to this. Sir, I have sent you 2 files. We just share with the group. So if you go here, actually you can mainly do 2 types of jobs. One is comparing the nucleotide sequences or DNA sequence, you can call it. Another one is a protein blast, where you can compare the protein sequences. So protein sequence comparison, you had a class or this is? No, but how to do the sequence alignment, multiple sequence alignment? No, No, nothing was selected. Okay, so you had already. Oh, okay. So then, suppose I have a protein here. I have sent a sequence that must have shared in your group. So you can take that sequence and then enter here. Oh, I think the copying is problem. And then if you just give a, make sure that it is a protein BLAST P, so that means it's against the proteins only. So what it does is it compares with already existing sequences, and then it tells you that whether the given sequence belongs to a particular organism which is already characterized. Apart from that, it will give you the closely associated sequences. Okay, so there are two types of alignments. One is a local and the global alignment. So the global alignment means it will try to match the whole sequence. So there will be a lot of gaps and other things would happen. Whereas the BLAST by default uses local alignment. That means if some of the sequences are locally similar to your reference sequence or the query sequence, then it will align with that and then show you. So of course we use both depending on what type of information you want to know. The local alignment is generally done when you want to compare it with the larger data sets. Say for example, I have given some, basically it's a yeast protein sequence. So, but I wanted to compare with all kinds of organisms, not they are closely related. Suppose it's unknown, then I just want to compare it with all, and then I want to know that which kind of a similar functionalities or domains can be there. I'm not interested in exactly how it is evolved and other things. Suppose if I want to study the evolution of particular protein, let's say that within the bacterial system, then what I need to do is I have to align the whole protein itself. So in that cases, you have to go for a global alignment. So basically, whether you want to go for a global or local depends on the type of sequence you want to compare. If you want to compare it with very closely associated organisms, obviously it should be a global alignment. But if you want to go for a vast database so that you get better information about the regions or the domains which are functionally important, then you can go for the local alignment. BLAST by default uses a local alignment because it's a data set of all the types of organisms. So, but if you specifically want to go for a global alignment, you can just directly type NCBI BLAST global alignment. It will open another page where you can see that. In fact, if I go back, so you can see here there is an option for global alignment. If you want to compare a particular protein associated, let's say that you have taken a protein, but you want to just compare that protein only with mammalian organisms, not with all the organisms, then obviously you would look for a global alignment. In that case, you can directly go for a global alignment. Rest of the procedures are going to be the same. Right. So now we have taken one protein and blasted it, and what we have seen here is that what type of similar proteins are already available in the database. So if you see here, you can see that it's 100% matching with, as I said, it's already a yeast protein and its function is also known. So this is basically a alanine transaminase. It's a class of transaminase protein. Apart from that, it will tell you that what are the other closely associated proteins from other organisms also. So obviously the yeast characterized would be very close to this one. If you go down, you may see other organisms as well. So since it's very common, so we are seeing most of the things within the yeast only. So first at 100, it comes as a yeast only. So if it is something of the rare protein, you may see from the other organisms as well. Okay. So of course you can go for further analysis based on the sequence and other things, which we will not go into that. But NCBI has a very good tutorial page. You can go through that and do that. But basic idea, of course, the same thing. If you don't have a protein but you have a nucleotide sequence, you can do it for the nucleotide sequence as well. So if you go to the home page of this one, you can see here. Just you have to choose whether you want to blast a nucleotide or a protein. That is a choice you have. Okay, right. So basic idea of BLAST is you have some sequence, you want to compare it with already reported known sequences. That is what it can do for you. All right. Okay. And another database is UniProt. So it's a protein database and it's very vastly used. So UniProt, generally the UniProt has a two types of databases. Instead of explaining it here, better we can go to the UniProt database itself. Can you just type UniProt database? Then it will take you to this page, so that I can explain you how it works. Rather than just giving a lecture, we can have a small demo. First, you can type it in Google UniProt. It's a web server, so you can directly open from your Google Chrome or Firefox. Okay, fine. So UniProt basically has two types of information. So basically UniProt was started as UniProt knowledge base, KB, that's what they call it as. But what has happened over the period of time is you submit a sequence. Suppose I have characterized a particular protein sequence. So I want to give it to the database. Obviously everybody wants to submit to the database so that it can reach the wider range of audience and it can be useful in many ways. So I want to submit it. It will be manually curated and then it will take it into the database. This was the very traditional way of pulling the data for a database. But after the genome sequence process has started, so instead of giving you a gene sequence or a protein sequence, people are started giving the genome. Okay, I think it will be done by that time. Okay, so people started giving a whole genome sequences. So now manually annotating this becomes a big challenge. So what people thought is we will use computational tools to annotate and get the gene sequences which is expressible and then which can be converted into the proteins, and those protein sequences would be given into the database. But there is a disadvantage here because you are not manually curating it, right? You are depending on the computational tool, and there is a possibility, there are two types of possible errors are that computational tools, of course it can be 90% correct, but sometimes it may predict the protein sequence wrongly. There is a possibility. So it needs to be manually curated further. And second thing is that once you give the genome sequences, there is a possibility that there are the same type of proteins, but with slight modifications like isozymes and all can also come into that one. So separating them by computational tools is a big challenge. But we don't want to miss that information because that information is huge than the manually curated information, right? So that's why UniProt came up with the idea that we will have two types of data in the database. Okay, so one is actually the UniProt KB, which they call it as UniProt KB sysprot. That means that the sysprot would be manually curated. That means once you publish a paper or if you submit in any database, okay, so they will check the nucleotide sequence, converting into the protein sequence, and then match with already existing sequence, whether it is a redundant or it is a new sequence. If it is a new sequence, they will assign an ID and then only they will add into the database. So this is manually curated. So you can see that, of course, manually curated may also have sometimes error. So, but you can presume that this is mostly error free. However, UniProt has TREMBL. TREMBL traditionally the gene sequence submission database, where it is computationally annotated and still that information is available here. So if you want a very particular protein sequence, you can go, but if it is not available in the manually annotated, computationally also it will show you, but it will tell you that it is computationally annotated. Then based on your own literature survey or your own studies, you can confirm that whether the sequence what it has given is correct, and then you can use it. So the only challenge here is that the number of sequences which are being submitted is so huge, you cannot do the manual curation. You can see the difference in the numbers. The manually curated sequence is something around 5 lakh or 5.7 lakh, you can see. However, computationally annotated is, I would say, something around 20 crore. So these number of sequences are yet to be manually annotated and brought into the database of Swiss Prot. However, it will take a very long time. Until that, we cannot ask the researchers, please don't use these many number of sequences for your research, right? That will drag back the research. Therefore, what people do is you just give it, but tell them that this is computationally annotated, so that you have to take an extra care when you use that sequences. That is the whole idea of UniProt having two types of data in that. Okay. So as on today, in total, of course, combining these two, UniProt was roughly 20 crore and 3 lakh number of protein sequences. Okay, you can call it as either manually annotated or computationally annotated. Okay, so we will quickly go through the UniProt, what are the different functions we can do it. So if you go for UniProt KV, it includes both the databases, but when it predicts the sequences, it will tell you that they are manually annotated or not. However, it has other functions also. I will not go into everything in detail, but I will just give you a brief idea. UniReference is a reference data where it has removed all the redundancies. So if you go to UniReference, any two sequences will not be exactly same. Okay, so that is what the UniReference, it's a reference sequence base, but number of sequences will be less, but you will be assured that these sequences, none of the two sequences are exactly repeating ones. And the UniPark is another database, it's a non-redundant, but per organism it will give you one simple sequence. Suppose you have an isozymes and all, but one representative sequence for every type of organism for a particular type of a protein would be given into that. So it depends on again what kind of a job you want to do. Suppose I want to functionally correlate my protein, I want to compare with the other proteins, I do not know what kind of function it may happen. So I can take UniPark and then or UniReference and then go for it. I don't want to compare it with all the data available. So I will just go with the reference database to do that. Or if I want to just submit a new sequence, I want to just make sure that the sequence has not been already submitted, then I have to refer with all the proteins which are submitted, then I will go for the whole proteins. Okay, right. So UniProt gives a unique ID for each and every protein sequence which annotates and updated in the database. Okay, I will just go with the one simple example, the transaminase example, and then explain you what kind of information you can get for that. Okay, so for example, we can take this particular protein, alanine aminotransferase. I have already given this ID, that is P52893. Okay. So I will just type P52893. Suppose if you have the name, suppose I know that it's a transaminase, then I can type transaminase from yeast. So you can use it something very similar to Google search. Of course, you cannot give that many of very non-scientific keywords, but about the protein what you are looking for. So for example, I am looking for a transaminase from yeast. I can type simply yeast transaminase. But it will give you many different strains of protein sequence, right? Yeast has different strains. So every strain sequence also it will give you. I want to go very specifically a particular strain, then I know the ID, I'm going for it actually. Okay. So if you go for the UniProt, what are the basic information you might get from the UniProt is, of course, the functional annotation, what type of function it does. So this protein is the alanine aminotransferase. So if you want to know that what actually you mean by alanine aminotransferases, they will give a schematic also, where what kind of a reaction it does, that would also be there. And then what's its taxonomy, since it's in yeast, how it is closely associated with the other proteins would be there. And then any post translational modification has been reported for this protein. and then how it is expressed in the system, in the biological system, what type of interactions it has. So you can go with all the informations. So here it is a taxonomy information, and this is any which site has been already reported for post-translational modification that can be done. Remember that these are all experimentally verified. It is not based on the computational predictions. Whatever the data you have here, it is experimentally verified. However, experimentally done, but some of them are computationally identified. That means, yes, this experiment has been done. This residue has been identified as a post-translational modification. Then that would have been computationally verified and then put it over there. Okay. So these are expression levels and what type of interactions it has, and its structure and family and domains. So the family and domains tells you that what kind of function it can perform, and then its complete sequence, and what are other similar proteins which are available, which are very close to this protein in the UniProt database. So that information also you can get from here. Okay, so mainly my topic is given to be around the structure. So I will just go to the structural tab and then explain you how do you assess the structural information in the UniProt. So that is what actually we are supposed to do. Okay. So there are two types of structures, as you know. One is experimentally verified, another one is computationally developed structures. Okay, I took this example very purposefully where the experimental structure is not available. Okay, suppose if it is experimentally available, it will tell you, it will take you to the protein data bank. For instance, I'm just typing one of the model proteins what we are using. Suppose I go for this particular protein. If I go to the structure, you can see here that there is a protein data bank PDB. So PDB is another database you might already know that it's a protein database where structurally resolved proteins are kept. Experimentally resolved structures are there. Okay, so in this particular protein, this is a different protein. So already experimentally resolved structures are available. So if I want to see that, I can go to the PDB, RCSB PDB, then I can get the structure of this protein, which is experimentally resolved structure. Alright. Okay, but this does not happen in the case of the protein. So here you can see the experimentally resolved structure. It's by NMR. We are not going into that details right now. But when it comes to this particular protein, if you go to the structure tab here, and then if you go down, you can see that there is no protein data bank information. Only it shows that either it is modeled with sysmodel or AlphaFold DDB. So that means this has only the computationally developed model structure. Okay, all right. So basically in the UniProt database, what you get is if you have any protein in your protein of your interest, you can type the protein's name with a specific organism's name, then you can get the sequence, functional domain, and either experimentally resolve the structure or computationally predicted structure of the protein that can be obtained from the UniProt database. So it is completely different from NCBI. Of course, these two are connected to each other. So if you have the UniProt ID, you can give it in the NCBI also, and it will retrieve the sequence. So it's not that they are completely independent, they are interrelated only. You can take this UniProt ID and you can keep it in the NCBI search directly. It will give you the proteins, all the kind of experimental information with citations from the NCBI also. So you can work in either of the way. Okay, so the important part is the protein structure prediction. That is what is supposed to be our major focus for today. So, so there are the challenges that you cannot resolve every protein structure experimentally. Of course, experimentally resolved protein structure has its own advantages and it's more reliable. That is different. But you cannot resolve timely the protein structures which you may want to predict it. So what kind of predictions, the protein structural predictions methods are available? The very first or traditional way of, very conventional way of predicting a thing is based on the homology modeling. Homology modeling is with the, if you have any doubt you can raise your hand. Okay, don't. Oh, I'm going fast. Oh, okay, fine. I thought I'm going slow. Okay, all right. So until here you are clear about the NCBI and UniProt, right? What it can do. Of course, it's a huge database, so you can keep exploring about it. Okay. So if you ask me, still I would say that still I'm also exploring the UniProt. Though we have been using it for last 10 years, but you go for a different protein, so you get a different protein, so you will look for a different functions, and ultimately this function, this kind of a functions you might have not worked earlier, and structural informations and domain assignments. So there are so many things are there in the UniProt. Okay, so but almost all experimentally annotated information is available at one place. That is the biggest advantage in the UniProt. And the NCBI, as I said, it is a very vast one. It works on literature, it works on the small molecules, if you are interested in drug design and all. And it has the information about the nucleotide and proteins as well. Okay, so this is very, I would say that this is very minimal introduction to the NCBI and the UniProt, because these are very vast databases, almost now running it for more than 25 years or even more than that. So you can imagine that how much of data has been put into that. Okay, so if you have any protein, it's better take the protein and then type the protein's name and organism's name and explore yourself. That is the only way you can understand the whole database. And of course, it's ever growing, so we can never understand the whole database in our lifetime. Better to focus on one particular protein and then look into its information. Right. As I said that our major focus for today is a protein structure prediction. Protein structure prediction. So we are not going to talk about how we experimentally resolve the protein. The challenge is that I know the sequence, but I have no clue about its function and I have no clue about its structure. And of course, structure functions are related. So if I know the functions, I can guess what could be the possible structure, or if I know the structure, I can possibly predict the function. Basically, that is the idea. So the very first idea of understanding the structure of a protein is partially to understand the function of the protein. So you may know, right? The basic fold, the topology of the proteins which are doing the same kind of functions would be same. Say for an example, if you take myoglobin from any organism, we are very sure that it is a helix bundle. So if it is a helix bundle with a heme, then you can say that it is possibly oxygen transporter. Right? So similarly, say for example, you have alpha beta alpha fold, then you can think that it could be potentially a DNA binding protein. So if you have a particular topology, you can at least guess what is its closest function could be. You cannot very accurately pinpoint, but at least, as I said, it's a DNA binding, it is an oxygen transporter. Such a kind of functional annotation also can be done if you know the structure of the protein. However, you cannot wait until these particular proteins of your interests structure is experimentally resolved. So that is the challenge. So can we computationally predict the structure of the protein? From there, can we understand the function of the protein? Now, of course, we can explore it further by doing the dynamics. How it is evolutionarily this protein has evolved, or how its function is different from its ancestors, or structurally how it is different from the ancestors. So those kind of comparative studies also can be done, provided you have a structure in your hand. It will take you to a completely different field of research if you know the structure. Okay, right. So basically the structural predictions are based on two types, though I have given in three because of the recent AI based ones, but largely it is of two types. One is the homology modeling, another one is ab initio. Ab initio you can call it as a de novo also. Sometimes they call it as a de novo or ab initio. Ab initio is basically from physics point of view. De novo generally they use it in biology. Okay, so I will first explain the homology modeling, which is traditional or conventional way of modeling the things. So in the homology modeling, what is our basic assumption is that if two proteins are homologous, then they should have a similar structure, because functionally they must be doing the same job. Therefore, their structural arrangement should be similar. This is our very basic assumption. So that means that if I want to, suppose I have a protein, only the sequence. I already told that if you ask me what I have in my hand is only the sequence of the protein. I don't have anything. Say for example, the transaminase what we have taken, that particular protein's sequence is known, but structure is not known. That is computationally predicted. Structure is not known. Okay, how they computationally predicted, that's what we are trying to understand. Okay, so I have the sequence in my hand. Now, what I have to find out is that, is there any other protein which could be potentially homologous to this? That means that that protein and my pairing or my reference protein, okay, could be performing a similar function. That's what we call it as a homologous proteins, right? When we say two proteins are homologous, your presumption is first that they evolved from a common ancestor to do the same function. Right? Then only you call it as a homologous proteins. When they have evolved from the same ancestor to do a common function, they must have retained their structural topology. There could be differences in the sequence, however, structurally they should be similar. So that is the basic idea of homology modeling. For that, suppose I have a sequence which I do not have a structure, but if any of the homologous protein from some other organism, if it has a structure, then I can guess that my structure should be similar to that, right? Because these two are homologous, right? So that is a very basic idea of homological modeling. So when you go for a homological modeling, where, as I said, the first assumption is that you have another protein which is homologous. What do you mean by that? These two proteins evolved from a common ancestor to do the same function in two different organisms. Therefore, they would share the same structural topology, right? Fine. So let us take this protein, transaminase. If you take transaminase, and then say for example, we can do the homology modeling in two ways. One is, these are two popular tools. There are a number of tools, of course. These are very popular and more reliable. I would say more reliable tools. I would explain a little later when do you say something is more reliable, because I cannot just say that this is reliable, this is not reliable on my own wish, right? It cannot be arbitrary. We will come to that little later. How do you say that it is reliable? Okay. So the modeler and Swiss model are most commonly used, more reliable and tested things. So I will not go into the modeler because modeler needs little amount of coding. You cannot submit it directly as a web server. You need to write some codes. So let us not go into the modeler, but it has its own advantages that you can explore it later. I have given the website this thing. I think they will share the slides also, so you can get the website and then you can explore. Swiss model is relatively easy. Easy means you don't need to do anything, just you give the sequence. So this is the Swiss model. I'm just going for the modeling here. I will give the same sequence because we do not have the same transaminase. I don't have a experimentally resolved structure I am giving here, and I ask you to search for a templates. Okay, I can give few options also here, whether my protein is a dimer, right? So if it is a homodimer, I can say that it's a dimer or it's a tetramer. Those information also I can give you here, so that it models as a dimer or a tetramer. That also you can do it. But this is a monomeric protein, it's known. So therefore you are giving it as a monomeric protein. So when I give this one, what it actually does in the background is, it goes to protein data bank, PDB, what we called it as a PDB, protein data bank. As I said, protein data bank is experimentally verified structures. So it does the alignment of my sequence with the proteins which are already available in the protein data bank. So if any of these two sequences are matching while aligning, match means it will not exactly match because I know that this is experimentally not resolved. However, if when it is aligned against the protein in the protein data bank, if the identity is more than 35%, then I can presume that these two proteins are okay. If it is more than 35%, then I can presume that that particular protein which is already available in protein data bank and my protein, these two are homologous. Okay, if it is less than that, probably they are not. Okay, so when I search only against the protein data bank, I am very sure that it compares with the experimentally verified structural proteins, right? So if I get something of that protein, let's say that I get a protein which is of 70% similar with the protein what I have in my hand. That means potentially these two can be homologous. The homologous protein which is available in the protein data bank is already the structure is resolved. Therefore, I would take that structure as a basic topology. What we mean by that is, suppose I am just drawing a randomly protein structure here. Okay, this is the protein structure resolved for the homologous protein, experimentally resolved. When I say homologous, what I mean is, I can take these basics. So this is the backbone, only the backbone I have drawn, right? Only the backbone amide bond. There are side chains, right? The side chains will be having different conformations. Some of them are exposed, some of them are buried, right? These are the side chains in the protein. When I compare these two proteins, what can happen? The backbone would adopt exactly the same arrangement. That's what we call it the topology of the protein. However, the side chains may be different because exactly not all the amino acids are matching with my template, which I call it as a template. Another one is the query. So my query has only 70% amino acids similar to the template. There is a 30% difference. What do I mean by this 30% difference? The side chains are different, right? The backbone is going to be same for your template and the query. Therefore, I can just hijack the basic topology from my template, and wherever my template and my query protein side chains are exactly same, those side chains I can assemble here. 30% of the side chains do not match. So those 30% of the side chains conformations, I have to get it and then put it here. It is something like playing the building blocks, right? I will give you an outline how it has to look like. But remember that what outline I am giving you has a different color, but what you have are different colors. 30% different in color, right? So what you do? First, wherever the colors are matching, you will first arrange that, right? Then what you will do, wherever the 30% is different, you will put your own logic to arrange that 30% into that one. So this is a very similar method only. It's just like playing a building block. So you just hijack the backbone, you are very sure that backbone is not going to change. So you take the backbone as such. Then side chains which are already identical, you take it from the protein data bank and fix it. Rest of the 30% you have to do that. So how do I know? Suppose say for example, I have a protein of 500 amino acid residues, right? It's around 525, let's say roughly 500 amino acids. When I say 30%, that means roughly 150 amino acids side chain conformations I do not know, right? So how do I choose their conformation? So what people have done is they have created a library. This library is very simple library. In the protein data bank, we have nearly 1 lakh proteins which are experimentally resolved. Right? In these 1 lakh proteins, you have only 20 amino acids. Right? Each amino acids, what are the different conformations? It has already adopted in the experimentally verified ones. Say for example, if I go for alanine, you have taken 1 or 2 conformations inside the protein. Right? When it is free in water, it's different. But when it's attached to the protein, it cannot have all the types of conformations. It will have 2 or 3 conformations. Say for example, tryptophan. Tryptophan can have only 5 different conformations in. Of course, we can rotate with respect to many atoms, but in proteins, so far only 5 different conformations only resolved. That means it must adopt one of these 5 conformations. So what I do is, I pick up those conformations and try to match here. How do I match in the sense? It should not clash with the neighbors. This is the first criteria. So I have, say for example, I take tryptophan. Tryptophan would be buried anyway, right? It's a hydrophobic, would have been buried. Suppose I had alanine, but in my protein it is a tryptophan. So now I have to remove that alanine and fix tryptophan. When I fix tryptophan, I have 5 options, right? From the library. How the library is made? From the experimentally known structures. From the library, when I took this one, and then that library is technically called as a rotamer library, because it's a different rotamers, right? So we call it as a rotamer library. So I try to fix one by one, whichever does not gives a very less clash with the neighboring residues. that would be the better conformation that protein can go. If you take any other conformation, see, it may clash with the neighbors. Right? So I would take the best conformation which has the very least clash with the neighbors, okay, and any potentially opposite interaction. Say for example, suppose I have a glutamate, okay, suppose I have to place the glutamate either this side or this side. Suppose there is another negatively charged residue, obviously I will not keep it there because there will be a repulsion. That means this glutamate should adopt a conformation which is completely exposed outside. Right? So this is basically a physics based model. There should not be a clash with the neighbor. There should not be any energetically repelling forces. Positive energy should not be created. I mean, in the sense that free energy should not be positive. Because if you have two positive charges or two negative charges, free energy is going to be positive. So that with that criteria, it will adopt the conformations from the set of library for those 30% of residues which are not available in my template. So that is how it builds a model. Okay, so say for example, this, please first leave this one. If you go for other models, you can see here that there are crystal structures which are similar here. So it tells you that, again, incidentally, this also has only 70% of, sorry, I think it is 46.54%. Okay, I think. So roughly it is, let's say 46% is identical with this protein, the protein which we have given. But it covers only from this residue. This part of the residues are not covered. That is called coverage. Coverage is very less. However, the protein has a similarity where once it covers. So based on that, you can create a model. Obviously, this region you do not have any protein to model. Therefore, this will be less reliable. Whereas this region is very similar to already experimentally resolved protein. Therefore, this region is more reliable. Okay. So you can build a model here. So if you say ask it to build a model, then it will build a model based on this. So it has already built a model. Okay. So now, what is the limitation of this method? The major limitation is that if you don't have a homologous protein, any homologous proteins which are structurally resolved, experimentally not resolved, then what to do, right? So that is where the problem. Of course, the model is generated, which regions are reliable, which regions are not reliable, those assessments you can do it here. There is a structure assessment. So the red regions are less reliable. And I think Ramachandran thought you know, right? So that tells you that how good your protein structure is. And you can see here that which are the regions which are predicted has a similarity and the deviations. So this will give you, based on the color, you can guess that which regions are more reliable, which regions are less reliable. So basically the red regions are less reliable, blue regions are more reliably predicted. Okay. But the major limitation, I will not say this is a disadvantage, but it's a major limitation is if any of the homologous protein is not known, then how do we model it? You cannot model it. As I said, that at least 35%. 35% is little poor, but still, suppose if, say for example, you are developed with 45% as a model, right? So that is okay. Then how do you solve that one? That is where the ab initio modeling comes in. So the basic idea of ab initio modeling is that you do not need to have a template. In the sense, you do not need to have a homologous protein to model your protein. Okay. So the ab initio models or de novo models works on two bases. Okay. One is physics based, physics and sequence based. We will come to that one. That is again a traditional way of solving the resolving. Another one is the deep learning and AI models. Okay. So I will not spend much time on physics and the sequence alignment based, but I will just introduce you. If you are interested, you can explore it further. So the physics based models, the most popular ones are ITASR and FIRE. Of course, there are different ones. The people who have worked on the AI models earlier worked on physics based models only. So as the AI has grown, then they shifted to this one, and this predictability become more reliable and popular. But earlier, David Becker, you know, right? So David Becker was awarded Nobel Prize in 2024 for AI model. He worked on RUSA. He is from the academic side, and there are other two who are awarded from Google Colabs. they are from the industrial side, the deep mind. So, so those two, John Jumper, so he is from DeepMind, that is from the Google. Basically, it was funded by Google. So these two were awarded Nobel Prize, two groups of people. So the DeepMind or Google's DeepMind is the one which developed the AlphaFold, which is based on AI based model. Whereas the David Becker also developed based on the deep learning based models. So that is a AI based model. But earlier, he has been working in this area for last 40-50 years. So if you see his works for maybe 10 years back, most of the things are based on physics based models or ab initio modeling only. His earlier career is on ab initio modeling. Okay. So there are two types, there are two popular softwares. As I said, again, there are so many, but these two are more reliable. So that is, one is iTaser and the another one is Spire. So I have given the links, you can go there and check. So if you want, the problem with these two is actually you have to register then use it. So of course you can register and you have to register with your academic mail ID. They will not accept your Gmail and other things. So any academic mail ID you can register, they will verify your affiliation. If it is for academic use, it's for free. Their idea is if you are using for any academic purpose and you are registering with any academic email ID, they will approve it in 1 or 2 days. It won't take much time. And then you can use it basically. So I will just quickly tell that what it does is you just submit the sequence, it will develop a model. Basic idea is that your sequence will be first bifurcated into smaller pieces and then compared with already a secondary structural prediction software. So the individual small segments will be compared with the already available secondary structures. Then the secondary structures will be assembled. Based on the secondary structures, tertiary interactions will be assembled. So basically, the information what you need is that what type of sequence would form what type of secondary structures, what is the probability basically, which probability is more, based on that it develops a model. So you do not need to have a homologous protein, you just give a thing, already they have a library, if you have these type of sequence structures, it will form this type of secondary structures. These secondary structures can be assembled only in this way to form the tertiary structures. So this is the basic idea of ab initio or de novo modeling earlier, and once the structure is modeled, they will it will create multiple structures, right? You cannot create one structure based on this, so it will create multiple structures. So every structure they calculate the potential energy. So which of these predictive structures have the least potential energy could be probably the most possible structures, right? Because more negative free energy means that's a more stable structure that particular sequence can adopt. So this is basically a sequence, very basically, it's a sequence alignment, but it is very local. Very small segments of sequence alignments you do. For the sequence alignment, you create multiple structures. From the multiple structures, you have to choose one structure that is based on the physics methods. Basically, the interaction free energies they calculate. Once the interaction free energies are calculated, whichever is giving the least free energy, that means more negative free energy, that would be possibly the correct structure. That is how it predicts. So here you don't need to look for a homologous protein. You just give the sequence, it will work in the background. That's all. Okay. So both iTaser and FireEye has small differences. However, the basic idea is this one only. So first assembling it is like sequentially constructing the things. So primary structures you give, they will construct the secondary structure, then tertiary structure, then optimize the structure, the overall structure. So that is the overall pipeline how it works. However, this ab initio modeling and again, basically the ab initio modeling again goes back to the sequence alignment. So you need to have a small segment of the sequence where it could match with already existing structures. So that is another challenge. And the predictability of this ab initio modeling for the vast number of protein sequences are time taking. As you increase the number of unknown protein sequences, it becomes less reliable. So this is the challenge. Okay, right. But still, it works fairly well. If you ask me 5 years or 7 years back, we all relied on this methodology only, including David Becker. Okay, so because you had no option. So this is the best at that point of time. I would say if it is 2010, I would have given the lecture on iTaser only. Okay, because that particular point of time, that is the reliable one. Then came this machine learning ones. So machine learning. Already you had some basics, right, in machine learning, right? So machine learning is basically statistics, right? Maybe only thing is what we do is basic statistics, but we have more amount of data. So if you ask me very basic machine learning things, if you want to do, if I give you 10 data points, you do it manually, right? If I give you a calculator and then if I give you or Excel sheet at 10 data points, whatever you can do in a machine learning, you would do it manually. Only thing is now I have 1 lakh or 2 lakh data points. So it becomes a challenge. Right? So that's why we rely on the softwares to do the things. That's the only thing. But the little advance of that one is the deep learning models. So the deep learning models are slightly different from the basic machine learning models. So the deep learning models, what it does is it creates the matrices of what are the different features or different types of, say for example, we will take some common example of the things. Say for example, I want to predict the salary of the people who work in AP government, not teachers, in AP government, that's all. So what are the features you think that, suppose what kind of a data if I collect I can predict it? So I don't know how it works. So it asks group A, group B, like there is a classification of group A or group B, right? Okay. So I can, so say for example, I want to predict that, say for example, how much cost sir will spend to buy his shirt. This is what I want to predict. Okay. Right. So now my only question is I want to do it for any employee who is working in AP government. So you give me the employee's data, I will predict you that how much he might spend on his shirt. So you think what are the things would decide that? Obviously his salary, but he will not tell me the salary, right? So then what to do? Then I have to go for, but sir will tell me his designation, right? And he will tell me that how long he is working. So I can guess his salary, right? Okay. So sorry sir, I don't know about your personal life, but I'm just taking it for granted. I'm sorry for it. Say for examples, let's say that sir has only one kid. Okay, if he is a boy, okay, he will not take a very costly shirt. I know because I have a boy. Okay, right. But if you have a girl child, you will take a costly shirt. It's not that your daughter chooses, right? So the feature includes your child and what is the gender of the child, right? Of course, 90% of the time it could be correct, right? So if I know sir's age, right, that also matters, right? So how long he is in service, because age and service period may vary. So if these are the information he is ready to give, right, you will not mind giving me this information. So if I know his age and if I know his what you call designation, how long he is in service, and from the personal life how many kids he has, out of that how many are boys and girls, and from their age how much he might spend on them, I can understand, right? Okay, then based on whether sir's wife is working somewhere or not, because that also matters. So whether she is working and how long she is in service and other things if he can give. Of course salary is not necessary, but how long she is in service, what is her designation, if that is there. If these features are known, and ultimately what I will do is I will take a data set of AP employees, not from sir, from other 1,000 people where I personally know them, how much they would spend on the structure. From their data I will create a model, because these are the properties. If these properties are there, how much he is spending on buying a shirt and what occasion, that also matters, right? So what occasion, right? So now what I will do is I will use that model, I will ask that questions to the sir will give me the answers for these, then I can predict that how much sir would spend on his shirt, right? So this is what actually the deep learning models improves it. Remember why it is little tricky is some of them are categorical data, right? Some of them are, say for example, sir has let's say only one kid, right? It's a categorical data, whether it is a boy or a girl, right? It's a categorical data. Some of them are numerical data. Say for example, how long he is in service. Yes or no, I cannot give you, right? So the traditional statistics has the restriction that the categorical data cannot be mixed with this. However, in the machine learning and deep learning, you can put these data as an input and then you can develop a model. So that is how do you do and all these things. We will not go into that, but what is the challenge you understood, right? Some of them would be, it's a binary data, right? When I ask her that how many kids, one kid or boy and girl, it's a binary. I can say boy yes or no. That means it's a girl. So it's a binary data, right? But when it comes to the service years, it's a 25, right? When it comes to years, it's a numerical, right? So these kind of, say for example, Sar has a car or a bike, right? Again, what is the cost of that? Because he must be spending fuel on that, right? So I indirectly want to calculate that. So whether he has a car or a bike and how much he is spending. If he says car, I will categorize into 3 or 4 classes, right? That becomes a categorical data, but not binary, right? Whether it is sedan, SUV, right, or MUV. So based on that, I can guess how much he would be spending on the travel, right? So these are the data actually which are from different types of data. The data type is different. In the traditional statistics, you will not preferably mix the data types, like categorical to numerical data, whereas here you are supposed to mix that and then create a model out of it. So that was the challenge people took, and that is what we extended to apply to the biological ones, where the deep learning models has come. Now, in the biology or the protein structure prediction, how does it work? What they do is create a graphical models that if these sequences are arranged in this way, how it would work. Okay, so one of the graphical neural network model, I will again not go into the proteins, which should be a little tricky, but a very most common example is your Google map. Okay, so I know all of you are familiar with Hyderabad, right? No? Okay, let's say that at least you know Hyderabad, right? So all of you must have come by train. So at least you know Hyderabad. So suppose if you have to drive to Hyderabad, what would you do? You will ask the Google map, how do I go, right? And you do put conditions on the Google map. You have to go with the fastest route. You want to go with the fastest route. How Google decide this is the fastest route? It's less traffic. But how does Google know that this is right now here it is less traffic? How many of you are using Google Map? You are actually not using Google Map. We are providing data to Google Map. Right? If 100 of people are in Gachibowli, switching on the Google Map, waiting to pass the Toli Chowki, the next immediate is the Toli Chowki. Google Map already knows that 100 of people are waiting at Gachibowli flyover to pass the flyover, because all of them are switched on the Google Map. So people have tried. One person took some 100 mobiles, switched on this. Okay. So virtually he hyped the traffic. Virtually, it's not actually a traffic. A single person putting all 100 mobile in. I don't know, remember the numbers, but he has shown that Google Map can predict wrongly, provided if you do this. Okay. That means that Google Map does not look into you. It just looks into your position from the mobile. So using the Google Map, you may think that what is advantage for the Google? They are taking the data from you. They are taking the traffic data from you. Of course, they are giving you back. Okay, this is how you have to travel. So let's say that there are 3 routes to Secunderabad. Right? Let's say this is UoH and this is Secunderabad. I'm sorry, my handwriting and drawing is very bad. Okay. So, but you can understand this is UoH, this is Secunderabad. Okay. Let's say that there is one route like this. There is another route like this. There is another route like this. There is another route like this. But ultimately they go to the Secunderabad. Okay. Now, how do I know that traffic is congested? Suppose the traffic is congested only, let's say that only in this route. Traffic is congested. So I can go through either this route, right? I can skip. I will not take this route or this route. So these two are not preferable, right? Because it will make me stuck there in the traffic. But I can choose the other routes. But how do you predict it? Mathematically. I told you the information, but how do you predict mathematically? It's a very simple graphic idea. So you segmented into some nodes, right? The points you have created, right? And you have connected the points. So that connecting the points will give you the distance, because the fastest route depends on the distance also, right? Now I have to choose between these two routes. At the same, these two routes are, traffic is less, but I have to choose the shortest among these two also. That is another challenge. I have to choose a shortest is easy. How many number of nodes are there? What is the distance between the nodes? Right? Nodes means every dots. What is the distance between these nodes which I have to choose? And which one will take me Secunderabad with the shortest route? Let's say that this one takes me with the shortest route. But how do you know? What they do is give a weightage. Suppose, how do you know that this one? Suppose 100 people are using Google Map here, 100 people are using Google Map, but 200 people are using Google Map only to cross this stretch. Right? Whereas when you take these nodes, only 20 people or 30 people are using Google Map who are crossing these stretches. Right? So what you do is, these are called edges. You weigh the edges. Right? 20 people are using here, 20 people are using here, 30 people are using here, 40 people are using here, 20 people are using here. Let's say that here also 20 people are using 30, 50, but in this stretch, let's say that some flyover construction is going on. 100 people are supposed to cross, but they are within that stretch for a very long time. Okay, but once they cross a given point of time, let's say within 15 minutes, only 50 people have used Google Map within this stretch. Right? Same way here 30, 20, 40, 20, 30. So obviously now I know that if I weigh the edges, how many people are within the nodes, how many people are stuck there for a long time using the Google Map. Right? So what it actually means for you is, this is the deep learning, that's all. Basically, of course, this is not deep learning, this is one of the deep learning, I would say. I cannot call it as, this ends here, this is one of the simple applications of the deep learning. So you understood, right? What is the basic idea, how we can, what has happened is, I use Google Map, I got the data, right? But now I have to tell you which route you have to adapt. So I generate the data, right? So that is the difference. One is, I collect the data and do the analysis, that is fine. I can collect the data and tell the traffic police, see there is a lot of crowd, you go and attend to that. That is not a generative, right? I am not going to generate, I just have a data. This data I give you the traffic police, they know that they have to deploy maybe 2 traffic constables here, maybe 4 constables here. However, they may need 10 constables here to clear the traffic. But I have not generated any data. Right? I have given what is available, you decide what you want to do. So that is one type of analysis. Another type is, I generate you the path, which is the shortest and less traffic path you can choose. This is called generative AI. Okay, right? So ChatGPT. Right? So what is the difference between Google and ChatGPT? You type, Google says that these are the places where the data is available. Now you go and read that. Right? That is not generative. Right? But what Gemini does, whatever the data is available, it combines for you and gives you a brief idea about how you can, what are the things you can understand from this data. Right? That becomes a generative. So the difference between the analysis and the generative is, you create a data, a new data set, but with the existing data only. Without existing data, this cannot be generated. If all of you switch off, let's say that government orders today saying that 2 days none of you should use Google Map in Hyderabad. Okay, then what will happen? Everybody will write to the higher authorities that I am not able to find a path to office, give me a leave, right? Because Google Map is banned here. Alright. So basically, if you don't give an input, Google cannot generate the output. This happens in a current mode, live mode, right? Every 5 minutes or 10 minutes, this data is kept updated. That is the challenge. Every 5 minutes, this data is very, very dynamic. It's not dynamic, it's very, very dynamic. Every 5 minutes, this data would keep changing. So your prediction also has to be every 5 minutes would change. So that is the challenge they face it actually. Okay. I mean, taking this to the biology is that what the interesting part is. Instead of these spots and that lines, can you imagine the amino acids in the spots and how these amino acids are connected to different proteins? Already you have 1 lakh proteins in your protein data bank, which is experimentally verified, right? And if you create a graph of all these proteins as a graph, what is the possibility that if these 2 residues are distanced by 2 residues only, 2 residues are 5 amino acids distance, amino acids in the sequence, but when it comes to the protein, what is the possible distance between them? If these 2 are 8 amino acids away from each other, what is the possible distance in the protein structure? Because even 8 amino acids, but they can come close to each other in the 3D structure. So if you can make a simple graph of all the proteins, which are already available in protein data bank, you can generate a model something like this with a weightage. If these 2 are amino acids and these 2 have certain distance, right? In some other protein, these 2 are little far away from that. Some other protein is little less distance from each other. So I give some average weight to that. So what should be the distance between these 2 amino acids? Like that, if you can create 20 cross 20 matrix for all the possible distances between the amino acids, that will be your input. That will be your input. It is just generating a data. Okay. From there, now as Google does, you can predict, if you give me a sequence, I will arrange it in the same order. Whatever already potentially the best possible way this can be arranged. Right? So basically it is a, maybe say, very high level of graphical neural network modeling. That is what AlphaFold does. Okay, I have made it super simple, I know that, but I have no other option how to explain it in more detail. Okay, so but the very basic idea is, you create a graph of each protein based on the amino acids. You say, for example, Kachiboli, Doli, Chowki, Magadipatnam, let's say, what do you call Agriculture and second Rabat. So let's say that these are amino acids. You have created such a kind of amino acids into your maps, graphical maps with the different nodes, points, and you will measure that what is the distance between them. Based on that, you do a weightage. You can generate a protein models, potential protein models from there. Here, the shortest route are your what you call your generative data, right? So you want the shortest route as a potential output, whereas here it will be what is the most possible distances found in the already existing data. That would be the condition to generate the data. Okay, so that is how AlphaFold. I mean, I will not say that this is how exactly AlphaFold works, but roughly this is how it can be interpreted. Very roughly, it's not very accurate way of describing AlphaFold. I know that it's a very, very super simple way of understanding how AlphaFold works. All right. Okay. In this way, what actually you need is a set of proteins which are already known. That's all. That is the only thing. Remember that this is a dynamic data, as I said, very dynamic data in the sense every 5 minutes it changes. Whereas in the protein structure, every 5 minutes protein will, if I give a sequence, you predict, you experimentally also determine the structure. After 2 years, it's not going to change. So actually speaking, that is less challenging than creating the Google map. Actually, Google thought that for Google, it is a simpler problem. Because they are creating, suppose we assume that every 1 year, same sequence of protein is adapting different structures in your body, it would not be a big computational challenge, like your traffic. So, but now you compare to the traffic map, or a traffic's graphical one, you take the protein, actually it is a simpler one. Because your protein, if I give you the sequence and create a graph, it is not going to change forever. So therefore, actually for Google, it's a simpler problem compared to the problems actually already. And that's why they took it and then they were able to succeed also. Okay. So what they did, okay, they did not stop there. So they created a model based on the already existing protein structures, and then they, if you give a sequence, it will generate. This would have not been very popular. Okay, there is a difference between somebody who acts well and then who becomes very popular, right? So, say for an example, who is earning more, Amir Khan or Salman Khan? No, by money wise, who is earning more? Salman Khan, but who is working hard? So becoming popular is different from working hard. Right? So that is what AlphaFold did. It's not about making the software and then do that. If you want to become popular, you have to fit something into the code. Right? So what they did was, there are 2 core sequences in the UniProt, right? For whether the experimental structures are available or not available, irrespective of that, they generated structure for all the sequences which are available in UniProt. Even you have an experimental structure, no problem. We are also generating one structure. This gives you a challenge. I have predicted a structure. Suppose 1 lakh out of this 2 crore, 1 lakh structures are already available in Protein Data Bank. Right? Now I am generating structures for 2 crore, including that 1 lakh. Right? I am giving you. You go back and check those 1 lakh structures which I have predicted with the experimental structures. It matches with the experimental structures. Right? So they had the courage to tell that I have predicted even the structure of experimentally known one. Go back and check. It 99% it matches with already existing structure. Therefore, I am able to generate 1 lakh structures with 99% accuracy. Now you accept rest of the 1 crore 99 lakh structure where you do not have an experimental data. Will you accept it or not? That is the challenge they put forth. And they have succeeded in that. Right? So therefore now what has happened is you go to UniProt, UniProt takes you to the AlphaFold and gives you the structure. Okay, so basically you can do it in 2 ways. I will just take only 3 minutes. Huh? Okay, so you can directly come from UniProt. Here it gives the AlphaFold database, right? So you can come here directly. So the predicted structure will be available here. If you do not want to go to UniProt, you have a UniProt ID. Okay, so you can go to the AlphaFold server. You just type AlphaFold server. It will take. I have given all the links in the PPT. They will share with you, and then you can view the sequence. Sorry. The UniProt ID, and it will give you the structure because they have generated structure for all the UniProt IDs. Okay, as and when the UniProt updates, AlphaFold also updates itself and predicts the structure. Now UniProt cannot escape. At certain point of time, what happened is UniProt said that whenever we submit a sequence, we will inform you. You please add the structures. Right? So that is what it is now interlinked. Okay, so at certain point of time, it becomes an inevitable tool. That's what ultimately it means. But we'll do a simple caution. Since it has been predicted in AlphaFold, it's not at 100% reliable. Okay, ultimately at the end of the day, it's a computational prediction. Okay, AlphaFold itself tells you that. Whenever you read Gemini or ChatGPT at the end of the line, okay, like a mutual fund, what they tell very fastly, right? In the mutual fund, it is subjected to the market risk, right? Same way, Gemini or ChatGPT says at the end that it may generate a wrong data. It is your responsibility to verify it. So AlphaFold does not leave the responsibility to you, but it gives you 2 important parameters. Okay, I will explain it very quickly. Okay, one is this confidence level. PLDDT, I have given the link to understand how it works and all. Remember that this PLDDT values based on the color, if it is blue color, it is very reliable. If it is cyan, it's acceptable, let's say. Whereas if it is orange and red, they are not reliable. In the same protein, some of the regions, it is not able to predict properly. This is not told by me. The AlphaFold itself claims that in some regions, I am not able to predict the structures properly. Therefore, if you are going to use this structure, you can use up to this sequence more reliably for any functional annotation. However, if you are doing any functional annotation based on these regions of the structures, sorry, I am not responsible for that. Okay, because these regions, we are not able to generate a reliable data. Basically, these sequence structures are very less found in the protein. That could be one reason, that the data is not available. Another important thing to understand is some of the proteins are structureless. there are something called intrinsically disorder proteins. So some of the proteins, very simple example is the protein which leads to the Parkinson's disease or the Alzheimer's disease. So the alpha-synuclein, these are P53 in cancer. Okay, these are all disorder proteins. They do not have a very reliable structure. They are dynamic in nature. So that could be one reason. Another reason is AlphaFold does not have enough data to generate that stretches of the sequences. So it could be either of the reason, but these regions are not reliable. So you have to be very careful when you take the structure from the AlphaFold, how reliable every region in your thing. Suppose your functional region is only this part, yes, that means that you can use this one to predict the functional orientation. Okay, and you can download the structure here from the PDB file and then you can use it. I'm supposed to take how to view the structures in PDB structures. I don't have time, but I just already Timon is installed for that purpose only. But I have things you can try it out. Let us have two options. Tomorrow morning I have a class. Either we will go for the Timon or we will go for machine learning for drug design. Don't say none of the above, please. Okay, that would be terrible option. So A and B only, no C. Both will not have time. See, even I cannot finish this one. Timer I have even not started. Yeah, I am okay with that if everyone is fine for that. Because I think 11 o'clock or 10 to 11:30. If I start timer, it will go for at least 45 minutes. So let us start with the timer. As much as possible, I will just try to cover it. Suppose you download this structure. If you are already doing this, just to download the PDB files and keep it on your desktop so that tomorrow we can straight away start the class. Already Timon is installed. So this PDB file you can download and keep it in your desktop. Tomorrow we will straight away start with Timon. I will try to cover it within 30 to 45 minutes. Maybe rest of the 45 minutes I will explain you how machine learning is used in drug designing. Yeah, that will be very brief. I will try to be brief on that. Okay. All right. So any questions? No. Okay. So then we will meet tomorrow morning 10 o'clock. Morning I will take 10 o'clock class. Dr. Manjari will take 11:30 class. Just to start the classes. So if you have any questions, in fact, you can write to me. No issues. I have given the mail ID also. They will share this one anyway. So if you have any specific questions, you can write to me. Yeah.
Sir, we have created the group, so I'll call out the names. You can collect your chart and sketch pens. Group 1 is T. Sambaswarao, Usharani, Gangaraju. So next group is K. Venkat Reddy, K. Lakshmi Kandama, M. Anjali. Next group is Dr. M. Ramakrishna, Dr. M. Anil Kumar, D. Satish. Is Divagar sir is there? Next group, Sunita Devi, Sindhu Priya, Sunita Raju, B. Teja. Next group is Sridevi, Prabhavati, Chandrakala. Last group is Dr. C. S. Srinivas Rao, Mr. M. Swamalu, D. Vasubabu.